It's time for another warm-up. Let's get going. First question here, which zoonotic bacterium causes each of the following conditions? So causes cat scratch fever, think of Bartonella. Causes Lyme disease, think of Borrelia burgdorferi. Causes recurrent fever from variable surface antigens, think of Borrelia recurrentis, and this is from ticks and lice. Causes bloody diarrhea, think of Campylobacter, so this is transmitted by puppies, livestock, and also by fecal-oral ingestion and even sexual transmission. Causes Q fever, think of Coxiella burnetii, so this is transmitted by spores from tick feces and cattle placenta. Yum. Causes tularemia, think of rabbit bites, and that's caused by Francisella tularensis. Causes leptospirosis, think of Leptospirilla species transmitted by animal urine. Causes cellulitis and osteomyelitis from cat or dog bites, think of Pasteurella multicida. All right guys, that brings us to the end of our warm-up. Let's get to that lecture now.
In this section of biochemistry, we're going to review energy metabolism, which covers how we convert the food we eat into energy. And we're going to start with glucose, which is one of the most important energy substrates. We'll talk about how we break down glucose, how we synthesize glucose, and how we store it as glycogen, and so on. Now, why do we need energy? Well, energy is required to drive chemical reactions. We call energy-consuming reactions endergonic reactions, and we call energy-releasing reactions exergonic reactions. For example, when you break a phosphate group off of ATP, that's an energy-releasing or exergonic reaction. Going from ATP to ADP releases a little energy, and then going from ADP to AMP releases energy, and so on. And we can use the energy released by these exergonic reactions to power energy-consuming reactions, or endergonic reactions. That's why ATP is so important for life, because it can power endergonic chemical reactions. So if ATP represents energy that our bodies can use, how do we make ATP? Now, you don't need to write all this down, but I want to give you a big picture overview of where we're headed. So we're going to work backwards from ATP. To make ATP, you need to add a phosphate group to ADP. In other words, you need to phosphorylate ADP. This is a process called oxidative phosphorylation. And we're going to couple that to the electron transport chain. We're going to go over that in another video. Don't really worry about it for now. Remember, we're just talking about the big picture. So again, working backwards, the electron transport chain requires some electrons. We get those electrons from NADH. NADH is an electron donor. Now when NADH donates an electron, it becomes NAD+. So for the electron transport chain to work, we need a steady supply of NADH electron donors. And that's one of the big functions of the TCA cycle. The TCA cycle also makes a little bit of ATP, but the main purpose is to generate NADH. And again, we're going to cover the TCA cycle in another video. Working backwards one more time. What do we need in order to keep the TCA cycle going? Acetyl-CoA. So as long as we have a steady supply of acetyl-CoA to drop into the TCA cycle, we can keep making NADH to feed the electron transport chain and make ATP. And acetyl-CoA comes from the decarboxylation of pyruvate. And pyruvate is generated from glucose through the process known as glycolysis. So that's going to be our focus for the rest of this video. How do we convert glucose to pyruvate? And it just so happens that glycolysis also produces a tiny bit of ATP and also a tiny bit of NADH. But the problem is that glycolysis takes place out in the cytosol, and the TCA cycle, the electron transport chain, takes place in the mitochondria. And NADH can't pass through the mitochondrial membrane. So there are a couple of different mechanisms to shuttle NADH and other electron donors into the mitochondria. The shuttle mechanism and the electron donor you use determines how much ATP will be produced. Oxidative phosphorylation and the electron transport chain use oxygen, and they produce a lot of ATP. But again, those processes take place in the mitochondria. And some cells don't have mitochondria, such as red blood cells. So red cells can't convert pyruvate to acetyl-CoA and run it through the TCA cycle and the electron transport chain. All red cells can do is glycolysis, which is anaerobic metabolism. Anaerobic metabolism is fairly inefficient. It produces a net of just 2 ATPs from each glucose molecule. So most of our cells metabolize glucose aerobically, using oxygen in the TCA cycle and the electron transport chain. And again, there are different shuttle mechanisms and electron donors involved. Cells that use a shuttle called the malate aspartate shuttle, such as the heart, the liver, and the kidneys, can generate a net of 32 ATPs for every glucose molecule that they metabolize. And cells that use a glycerol 3-phosphate shuttle, such as the brain and skeletal muscle cells, can only generate a net of 30 ATPs, which is still really good, but it's just a little less than the malate aspartate shuttle. Before a cell can begin to metabolize glucose, it has to transport that glucose from the circulation into the cell. Now, this occurs via one of several different types of glucose transport proteins, which are called GLUT proteins, or glucose transporters. There's GLUT1 transporters, which are found on red cells and also in the endothelium of the blood-brain barrier, in addition to other tissues. But GLUT1 is the transporter responsible for low-level basal glucose uptake, which allows cells to take in glucose regardless of whether or not insulin's around. Remember, cells like red cells have to have glucose. That's the only energy metabolite that they can use. So they have to be able to take up glucose even without insulin. GLUT2 transporters are found on cells that regulate glucose, such as hepatocytes and pancreatic beta cells. Although there's some evidence that on beta cells, GLUT1 may actually be more important than GLUT2 for stimulating insulin release. GLUT3 transporters are found mainly on neurons and also found on the placenta. GLUT4 transporters are found in skeletal muscle and adipose tissue. And these are the transporters that require insulin. This is the insulin-dependent glucose transporter. And then the GLUT5 transporter is responsible for fructose uptake, especially in the GI tract. After glucose has been transported into the cell, you need to phosphorylate the glucose to trap it inside the cell. So you add a phosphate to glucose so that it becomes glucose 6-phosphate inside the cell. And then once it becomes glucose 6-phosphate, it's trapped in that cell. And we've said that enzymes that phosphorylate things are called kinases. And there are two enzymes that can phosphorylate glucose and other 6-carbon sugars like fructose: hexokinase and glucokinase. So let's start with glucokinase. Glucokinase is unique to cells that regulate glucose, just like we saw with GLUT2 receptors. So glucokinase is found in the liver and also in the beta cells of the pancreas. Now, glucokinase has a very high Km, which means it needs a lot of substrate to hit a decent velocity reaction. So you need a lot of glucose around in order for glucokinase to work. But it also has a very high Vmax, which means that once it does start working, it works very quickly and very efficiently. So when there's a lot of glucose around, it makes sense to have an enzyme that can handle that large amount of glucose. When there's not much glucose around, you don't want the hepatocytes to take up glucose and store it. You want them to share the glucose with other tissues. So that's why a high Km is very important for glucokinase in the liver. Now you can use the L in glucokinase and the L in liver to help you remember that glucokinase is found in the liver cells. Now what about hexokinase? Hexokinase also generates glucose 6-phosphate, but it has a low Km and a much higher affinity for glucose. In other words, the concentration of glucose doesn't have to be very high for this enzyme to work. Even when glucose levels are very low, hexokinase will be able to trap that glucose that is there, trap it in the cells by phosphorylating it. And it has a low capacity and a low Vmax. And hexokinase is not induced by insulin, whereas glucokinase is induced by insulin. So again, the first step of glycolysis is to phosphorylate glucose to G6P. Let's go over the glycolytic pathway, which is found on the right side of this diagram in your study guide. You do not need to worry about memorizing this entire pathway, but there are some key steps and enzymes that you do need to know. The first step is letter A, is either hexokinase or glucokinase, which makes letter B, glucose 6-phosphate. Then glucose 6-phosphate goes to letter C, which is fructose 6-phosphate. And that's converted to letter D, fructose 1,6-bisphosphate, so 2 phosphates. And the enzyme that does that's a very important enzyme. It's phosphofructokinase 1, or PFK1, which is letter E. Now PFK1 is important because it's the rate limiter of glycolysis. It's very important to know the rate-limiting enzymes in these pathways. So certain steps in this pathway are very tightly regulated, and this is one of them. You need to regulate when glycolysis is and isn't taking place. So when would you want glycolysis to take place? You would want cells to be able to break down glucose and generate ATP when they're starving for energy. So AMP is a molecule that will stimulate PFK1. Now letter F is AMP. When there's a lot of AMP around, that means you've already broken down ATP to ADP and then ADP to AMP. So AMP is a signal that there isn't much ATP around, and that stimulates PFK1. And then letter G is another molecule that stimulates PFK1, which is fructose 2,6-bisphosphate. So G is fructose 2,6-bisphosphate. We're going to talk about that a little bit more later. Then the other way to regulate PFK1 is to inhibit it. So what's going to inhibit PFK1? Well, high energy states. So letter H is ATP. If there's already a lot of ATP around already, you don't need to make more of it. Another thing that inhibits PFK1 is citrate, which is letter I. So citrate is a substrate of the TCA cycle. So if you're overwhelming the TCA cycle, there's a huge amount of extra citrate around that's going to cause a feedback inhibition of PFK1. Then the next few steps of the pathway have nothing for you to fill in. Fructose 1,6-phosphate, which was letter D, gets split into two things, glyceraldehyde 3-phosphate and dihydroacetone phosphate, or DHAP. But DHAP can be converted to glyceraldehyde 3-phosphate, which means that for every glucose that you started with up at the top, you get to go down the bottom half of this pathway twice. And take note, you have to use energy to add phosphate on two of those previous steps. And now that you're splitting that 2-phosphate sugar into two different molecules, and in the second half of the pathway, each time you go through it, you're going to generate 2 ATPs. So you put in 2 ATPs, but you ultimately get 4 ATPs out. So that's why we said that anaerobic metabolism generates a net of 2 ATPs. So moving on down the pathway, you go from glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate to 3-phosphoglycerate to 2-phosphoglycerate, and then you get to letter J, phosphoenolpyruvate, or PEP. And the enzyme that converts PEP to pyruvate is letter K, pyruvate kinase. Now this is another heavily regulated step. The substance that stimulates pyruvate kinase is fructose 1,6-bisphosphate. That's letter L, fructose 1,6-bisphosphate. We saw that earlier as substrate D. So an upstream substrate is stimulating the downstream mechanism, which makes sense because you don't want these substrates building up. And then letter M is ATP, and then N is alanine. And again, these ATP and alanines are going to indicate that you already have a lot of energy. You don't need to make any more pyruvate. So that's going to inhibit pyruvate kinase. So again, the key enzymes that you need to know are A, E, and K. Letter A was hexokinase and glucokinase. Letter E was PFK1, or phosphofructokinase 1, and letter K was pyruvate kinase. So those are the three main glycolytic enzymes important for you to know for your test. We're going to look at the gluconeogenesis side of this diagram in another video. Let's think about what happens to someone who's deficient in one of these glycolytic enzymes, which means they can't perform glycolysis. These patients are going to present with hemolytic anemia. So think about it. Red cells don't have mitochondria, so they have to be able to metabolize glucose in order to make ATP. So if there's a problem with this glycolytic pathway, the red cells can't make ATP, so the sodium-potassium ATPase won't work, and that means that they can't regulate sodium and potassium homeostasis, and the cells are going to swell and then lyse. And as red cells lyse, that results in hemolytic anemia. So what causes this? Well, pyruvate kinase deficiency is the most common glycolytic enzyme deficiency, and that's the one you should know for your test. Pyruvate kinase deficiency. Look at the next question. A muscle biopsy reveals elevated glycogen levels, elevated fructose 6-phosphate, and decreased pyruvate. What's the enzyme deficiency? Well, because pyruvate is decreased and because glucose is being stored as glycogen, you know this is a problem with glycolysis. So you have to think about which of your glycolytic enzymes is deficient. Because you have an elevated level of fructose 6-phosphate, this suggests a deficiency of the enzyme that converts fructose 6-phosphate to the next substrate. So what enzyme was that? PFK1. So the answer here is PFK1. Let's talk about how our bodies regulate this complicated glycolysis pathway. We saw earlier that fructose 2,6-bisphosphate was one of the things that can stimulate PFK1 and thus stimulate the glycolysis pathway. Well, where does fructose 2,6-bisphosphate come from? Fructose 6-phosphate can either be converted to fructose 1,6-bisphosphate by PFK1, which is the normal glycolytic pathway, or fructose 6-phosphate can be converted to fructose 2,6-bisphosphate by an enzyme called PFK2. Not PFK1, but PFK2. So PFK1 makes fructose 1,6-bisphosphate, and PFK2 makes fructose 2,6-bisphosphate. It's not too hard to remember. Now, when you eat, when you're in a well-fed state, your body's going to release insulin. And insulin's going to stimulate PFK2 to make more fructose 2,6-bisphosphate, which in turn will stimulate glycolysis by stimulating PFK1. So insulin does not stimulate PFK1 directly, but it does stimulate glycolysis by stimulating PFK2. Now contrast this to the fasting state. When you're fasting and blood glucose levels are low, you don't want to stimulate glycolysis and break down glucose. You want the liver to synthesize glucose through gluconeogenesis. So in the fasting state, instead of releasing insulin, our bodies release glucagon. The actions of glucagon are the opposite of the actions of insulin in a lot of ways. So instead of stimulating PFK2, glucagon stimulates an enzyme called fructose bisphosphatase 2, or FBPase 2, which converts fructose 2,6-bisphosphate back into fructose 6-phosphate, which can then be used for gluconeogenesis. So glucagon stimulates the conversion of fructose 2,6-bisphosphate to fructose 6-phosphate. That's going to shift everything toward gluconeogenesis to raise blood glucose when you're fasting. Now, there's one cool little wrinkle that may just blow your mind. PFK2 and FBPase2 are both part of the same enzyme complex. It's one enzyme with two separate functional domains. And whether or not the enzyme is phosphorylated determines which domain is active. And it's the presence or absence of glucagon and insulin that determine which functional domain is operational. The next study guide question says, how does a low insulin, high glucagon state inhibit glycolysis and lead to conversion of energy? So this is getting into the nitty-gritty of how the presence of insulin or glucagon can switch this enzyme on or off. So what happens is that when you are fasting and blood glucose drops and glucagon rises, you have a low insulin to glucagon ratio. So glucagon binds the glucagon receptor, which is a G protein linked receptor that stimulates adenylate cyclase. Well, what does adenylate cyclase do? It converts ATP into cyclic AMP. So then cyclic AMP is going to activate protein kinase A, which phosphorylates the enzyme FBPase2 and activates it. So when you're in the fasting high glucagon state and protein kinase A has been activated, this enzyme complex is phosphorylated, and that turns on the FBPase2 domain and turns off the PFK2 domain. So in the fasting state, you're going to drive the reaction toward more fructose 6-phosphate and toward gluconeogenesis. But in the fed state, protein kinase A is inactive and the PFK2 domain is active, so that drives this reaction toward fructose 2,6-bisphosphate and glycolysis. And to review, how does fructose 2,6-bisphosphate stimulate glycolysis? It stimulates PFK1, which converts fructose 6-phosphate into fructose 1,6-bisphosphate. All right, I know this is complex. A lot of these enzyme names sound really similar, and the regulation just isn't easy. So make sure you take some time drawing those pathways and thinking through this. You can do it. But for now, let's pause the video and work through the end of session quiz. First question, what enzymes convert glucose to glucose 6-phosphate? Well, glucokinase in the liver and the beta cells, and hexokinase everywhere else. Next, what is the clinical consequence of a glycolytic enzyme deficiency, such as pyruvate kinase deficiency? That's going to result in hemolytic anemia. The red cells can't generate ATP, so they can't maintain the sodium-potassium ATPase, and that means the red cells swell and lyse. Next, what enzymes are responsible for increasing and decreasing the intracellular levels of fructose 2,6-bisphosphate? Well, there's phosphofructokinase 2, which increases levels of fructose 2,6-bisphosphate, and fructose bisphosphatase 2, which decreases fructose 2,6-bisphosphate. And the last question, what's the rate-limiting enzyme in the glycolytic pathway? It's phosphofructokinase 1. That's it for now. I'll see you next time.
It's time for another warm-up. Let's get going. Damage to which part of the basal ganglia produces hemibolismus? So this is the subthalamic nucleus, which can be lesioned in a lacunar stroke, and then you will have too much movement, which will present as that hemibolismus. Next, what are the retroperitoneal abdominal structures? Well, we use a couple of mnemonics for this one, like sad pucker or a duck pair. We favor a duck pair. The A is for adrenal glands. D is for duodenum, the second, third, and fourth parts of the duodenum, but not the first part of the duodenum. U is for the ureters. C is for the colon. This is the descending and ascending colon, but not the transverse colon. K is for kidneys. That's the duck. And then for pair, P is for the pancreas, everything except for the tail of the pancreas. E is for the esophagus, at least the part that's south or below the diaphragm. And then obviously the upper esophagus is in that mediastinum. A is for aorta, and then R is for rectum. All right, that brings us to the end of the warm-up. Let's get to that lecture now.
In the last video, we talked about glycolysis. And before we move on to the TCA cycle and the electron transport chain, I want to cover another important process related to glucose metabolism, which is gluconeogenesis. Hang on. Something's not quite right. Hey, this is not my Biochem shirt. That's more like it. Now, where was I? Oh yeah, gluconeogenesis. Gluconeogenesis is the process of synthesizing glucose, mainly in the liver, so that it can be exported to the rest of the body when the blood glucose gets low. Now, in your study guide, you see that the gluconeogenesis pathway is similar to the glycolysis pathway. In glycolysis, we're starting with glucose and converting that to pyruvate and ultimately lactate. But gluconeogenesis is basically the same thing, but in reverse. So instead of going down the pathway, now we're going up from pyruvate toward glucose. The reason that the gluconeogenesis pathway is a little different is that you have to overcome the 3 steps of glycolysis that are irreversible. So you can't use those same enzymes. Steps that are reversible steps can share enzymes for gluconeogenesis, but the irreversible steps need different enzymes to go the other way. So down at the bottom, the conversion of phosphoenol pyruvate to pyruvate is irreversible. So to work around that, you use the enzyme O, which is pyruvate carboxylase. And pyruvate carboxylase requires the coenzyme biotin, which is letter P. So carboxylation, or adding CO2 to something, requires biotin. So pyruvate carboxylase requires biotin. That's going to create letter Q, which is oxaloacetate. Letter R is acetyl-CoA, which is stimulating this process. So remember, acetyl-CoA is what your body's feeding into the TCA cycle. So if there's an abundance of acetyl-CoA around, that signals that you have plenty of energy around, and you need to go back up this pathway. Then next, to get from oxaloacetate to PEP, you use enzyme S, which is PEP carboxykinase. So PEP carboxykinase takes oxaloacetate and makes PEP. Then you use the reversible enzymes to go back up the pathway on the right until you come to phosphofructokinase 1, which again is irreversible. So you have to use a different enzyme to get past that step, which is letter T, fructose 1,6-bisphosphatase. And that's the rate limiter of gluconeogenesis, fructose 1,6-bisphosphatase. Now, fructose 1,6-bisphosphatase is stimulated by ATP, which is letter U. So again, if you've got a lot of energy around, you can undergo gluconeogenesis. You don't need to be selfish with the glucose. You need to be sharing more glucose. Then letters V and W are things that inhibit this enzyme. So letter V is AMP, and letter W is fructose 2,6-bisphosphate. Again, it makes sense. These are molecules of energy deficiency. So if you're energy deficient, you don't need to be sharing glucose. And if you peek over at the glycolysis pathway on the other side, you'll see that when you reverse the ratios so that you have low ATP and high AMP and high fructose 2,6-bisphosphate, that's going to stimulate PFK1 and drive the pathway in the other direction toward glycolysis. Then letter X is the enzyme glucose 6-phosphatase, which converts glucose 6-phosphate into glucose. So there are 4 unique enzymes in the gluconeogenesis pathway: pyruvate carboxylase, PEP carboxykinase, fructose 1,6-bisphosphatase, which is the rate limiter, and glucose 6-phosphatase. So make sure you know those enzymes. Now, where is gluconeogenesis actually taking place? Well, it's a very specialized process with these very specialized enzymes. So only cells that are designed to share glucose are going to have these enzymes. For instance, the reason that muscle cells can't perform gluconeogenesis and make their own glucose is that the muscles don't have glucose 6-phosphatase. So gluconeogenesis occurs mainly in the hepatocytes. These specialized enzymes can also be found in the kidney and in the intestinal epithelium. And when does gluconeogenesis take place? Well, it takes place whenever blood glucose is low. So when your blood glucose is low and your cells need glucose, hepatocytes start doing 2 things. Number 1, they're going to start sharing their glycogen stores, which we're going to go over in the next video. And number 2, the hepatocytes will start converting other molecules into pyruvate and using that pyruvate to make glucose. And that's gluconeogenesis, the making of new glucose. So if you're unable to create new glucose from other substrates, the blood sugar is going to remain low and you're going to get hypoglycemia in the fasting state. So what specifically can enter this process of gluconeogenesis? Again, it's anything that can be converted to pyruvate. So you have odd-chain fatty acids that generate propionyl-CoA, and that propionyl-CoA can ultimately be converted to pyruvate, which can then be used for gluconeogenesis. A lot of your TCA cycle molecules can be converted to pyruvate and used in gluconeogenesis. Some of these molecules can be converted to oxaloacetate, which can then enter this gluconeogenesis pathway. Also, some amino acids can be converted to TCA cycle substrates, or to oxaloacetate or to pyruvate, which can then be used for gluconeogenesis. Have you ever stopped to think about why our bodies go to such great lengths to make sure glucose is available? I mean, if there are all these other things that could be made into pyruvate and acetyl-CoA and then run through the TCA cycle, why bother with gluconeogenesis at all? What's so great about glucose? What's wrong with using fats and proteins and other stuff for energy? Well, it has to do with the amount of energy the different energy molecules can release. And that brings us to Gibbs free energy, which is very testable and actually does show up on step 1. So look at the question in your study guide. What is the formula for Gibbs free energy? The equation for Gibbs free energy is delta G equals delta H minus T times delta S. So the change in G equals the change in H minus T times the change in S. So G is Gibbs free energy, H is enthalpy, or the heat change in a constant pressure reaction, T is temperature, and S is entropy, which is disorder and randomness. Now remember, we said we have to couple energy-consuming endergonic reactions to energy-releasing exergonic reactions. And some compounds have more energy-releasing potential than others. So if you compared a molecule of glucose to a molecule of ATP, which of those has more exergonic potential? Well, glucose, of course, because you can generate around 32 ATPs from just one molecule of glucose. With ATP, you're just going to break off each of those phosphate groups and release energy that way. And here you see the relative exergonicity, or the Gibbs free energy release of certain compounds. So phosphatidylpyruvate can release 62 kilojoules of energy per mole, ATP can release 31 kilojoules, and AMP can release 14 kilojoules. So you can see here that as you go through the glycolytic pathway in the TCA cycle, you're actually losing some energy along the way. You're becoming less efficient, and you're generating compounds that are less and less exergonic. So glucose can release more energy than PEP, and PEP can release more energy than plain old pyruvate. And then ATP is what you're ultimately trying to generate. So be aware of the relative exergonicity of these molecules. That's pretty much all you need to know about Gibbs free energy. Since we have a little extra time, I want to review some of the important rate-limiting enzymes that we've already come across, and a few that we haven't gotten to quite yet. So remember, rate limiters are very important to know for step 1. And actually, let's do this as a whiteboard review. So over here on the right, I've listed several important rate-limiting enzymes, and we're going to match them to the biochemical processes on the left. The rate-limiting enzyme in de novo pyrimidine synthesis was carbamyl phosphate synthetase 2. Not CPS1, it's CPS2 for pyrimidine synthesis. And for de novo purine synthesis, it was glutamine PRP amidotransferase. The rate limiter for glycolysis is phosphofructokinase 1, PFK1. For gluconeogenesis, it's fructose 1,6-bisphosphatase. For glycogen synthesis, which we'll cover in the next video, the rate-limiting enzyme is glycogen synthase, which is pretty easy to remember. And then for glycogenolysis, where you're breaking down glycogen to release glucose into the bloodstream, the rate-limiting enzyme is called glycogen phosphorylase. Then after glycogen, we're going to talk about the TCA cycle, and the rate limiter in the TCA cycle is isocitrate dehydrogenase. And then for the hexose monophosphate shunt, the rate limiter is glucose 6-phosphate dehydrogenase, or G6PD. So that's a little preview of coming attractions for the next few videos. And for your prize, here's a groovy little psychedelic smiley face. Far out. Now it's time for the end of session quiz. So pause the video and work through the end of session quiz questions, and then we'll go through the answers together. First question, what irreversible enzymes are involved in gluconeogenesis? You have pyruvate carboxylase, PEP carboxykinase, fructose 1,6-bisphosphatase, and glucose 6-phosphatase. Next, what enzyme catalyzes the rate-limiting step in gluconeogenesis? That's fructose 1,6-bisphosphatase. And the last question, order the following molecules by how much energy they contain that can be made available to fuel endergonic reactions. So from the highest energy to the lowest energy, it's glucose, then pyruvate, ATP, AMP, and then adenosine. That's it for now. I'll see you next time.
Hey man, how's it hanging? Studying hard? Groovy. Ready for a little study partner quiz action? Here goes. What's the formula for Gibbs free energy? Right on, man. Delta G equals delta H minus T times delta S. Far out. Free energy, man. It sounds so groovy, but it's not just like positive vibes and stuff. It's the amount of energy released by an exergonic reaction. But who's this Gibbs guy anyway? Energy doesn't belong to anybody, man. It's free. It belongs to, like, the cosmos. Far out. Whoa, dude, is that a double rainbow?
It's time for the 3-question warrant for Biochem 9. Let's get going. Which cytokines inhibit Th1 cells? That's going to be interleukin 10, and that's going to be what's inhibiting those Th1 cells. Next, outline the flow of aqueous humor. So this formed in a capillary bed in the ciliary body, and that's going to go further on and be secreted into the posterior chamber. Then it flows between the angle formed by the lens and the iris diaphragm. Then it moves into the anterior chamber and is finally reabsorbed by the canal of Schlemm. Next question, what is the mechanism of action of metoclopramide? So this is an antagonist at the dopamine D2 receptor. It's also a serotonin agonist, so it will increase contractility in the GI tract and move things along. All right, that's it for the warm-up. Let's get to that lecture now.
We've covered glycolysis and gluconeogenesis, and there's one more big thing you can do with glucose, which is to store it as glycogen. Now, glycogen is a huge polysaccharide made up of glucose molecules, which is how our hepatocytes and muscle cells store excess glucose for use later when you're fasting and blood glucose levels start to drop. The process of making glycogen is called glycogenesis. So let's look at the glycogenesis pathway in the study guide. Glycogenesis begins with glucose, and you have to trap it in the cell by phosphorylating it, by adding a phosphate group to make glucose 6-phosphate. The two enzymes responsible for that were glucokinase and hexokinase. And do you remember which enzyme was found in which tissues? Glucokinase is found mainly in the liver and the beta cells of the pancreas, and hexokinase is found in other tissues. So if we're talking about glycogenesis in skeletal muscle cells, it starts with hexokinase. So these enzymes turn glucose into glucose 6-phosphate, and then from there, you're going to move the phosphate group from the 6th carbon to the 1st carbon to make glucose 1-phosphate. This is different than glycolysis. With glycolysis, we went from glucose 6-phosphate to fructose 6-phosphate. Here, to synthesize glycogen, we're going from glucose 6-phosphate to glucose 1-phosphate. The next step is to convert it to something called uridine diphosphoglucose, or UDP glucose. And this is catalyzed by an enzyme called UDP glucose pyrophosphorylase. It's going to take that glucose 1-phosphate and make UDP glucose. So it's priming the glucose to be attached to a larger molecule. And the enzyme that attaches UDP glucose to the expanding glycogen molecule is called glycogen synthase. This is the rate-limiting enzyme for glycogen synthesis. Glycogen synthase is going to make what are called alpha-1,4 glycosidic linkages, which make the chain longer and longer and longer. There's another enzyme that makes branches in the glycogen chain, which is called branching enzyme, and it makes alpha-1,6 glycosidic linkages. Glycogen can get to be rather enormous, so the branches help you keep it somewhat compact. So you use alpha-1,4 and alpha-1,6 linkages. So that's glycogen synthesis. The purpose of glycogen is to store glucose for later. When you need glucose, you're going to start breaking down that glycogen and releasing the glucose. And that's a process called glycogenolysis, lysis of glycogen. So take a look at the diagram in the study guide. To break down glycogen, you either have to break down the alpha-1,4 linkage at the top of the diagram, or you have to break down the branching alpha-1,6 linkage at the bottom left. Let's start with the alpha-1,4 linkage. The majority of glycogen is composed of these alpha-1,4 linkages. And the enzyme that's used to break down the alpha-1,4 linkage is called glycogen phosphorylase. So glycogen phosphorylase breaks down the alpha-1,4 linkage and cuts loose one glucose 1-phosphate molecule. Then an enzyme called phosphoglucomutase converts that glucose 1-phosphate to glucose 6-phosphate. And then the enzyme glucose 6-phosphatase converts it back into glucose. Now, where do we see glucose 6-phosphatase before? Well, that's the last step in the gluconeogenesis pathway. And here we see that it's also the last step in glycogenolysis, converting that trapped glucose 6-phosphate back into glucose. Now remember, we said that muscle cells don't have this glucose 6-phosphatase enzyme. That's one reason muscle cells can't perform gluconeogenesis. And it also means that while muscle cells can break glycogen down into glucose 6-phosphate, that glucose 6-phosphate is still trapped in the cell. So it's going to be used by the muscle cell to generate energy through glycolysis. So the only reason a muscle cell breaks down glycogen is for its own use. Muscles can't share energy. Sharing energy is the liver's job, and hepatocytes do have glucose 6-phosphatase. So those are the 1,4 linkages. Now what about the branching linkages, the alpha-1,6 linkages? The enzyme that breaks those down is alpha-1,6 glucosidase, which is also referred to as debranching enzyme. So debranching enzyme, or alpha-1,6 glucosidase, breaks down the alpha-1,6 linkage directly into glucose. You don't have to go through all those other steps with the alpha-1,6 linked glucose. So it's a little bit more efficient. And actually, debranching enzyme has two functions. Let me illustrate it for you. This is your branched glycogen molecule, and glycogen phosphorylase is cooking along, breaking off glucose 1-phosphate molecules one at a time. But when it gets close to a branch point, one of those alpha-1,6 linkages, specifically when it gets exactly 4 glucose molecules away, glycogen phosphorylase stops working. That's when debranching enzyme goes to work. Like I said, debranching enzyme has two functions. One function is called a transferase activity. Specifically, it's called 4-alpha-D glucanotransferase activity. This means that debranching enzyme can take 3 of those last 4 glucose molecules and transfer them to another chain. So transferase transfers the branch to another chain so that glycogen phosphorylase can keep working on it. Then the other function of debranching enzyme is the alpha-1,6 glucosidase activity, which we already discussed. That's how debranching enzyme removes that last glucose molecule. Now let's talk about the regulation of glycogenolysis for a second. We need to regulate the processing of glycogen for the same reasons that we needed to regulate glycolysis and the reasons we needed to regulate gluconeogenesis. Basically, you don't want to use your body's energy to break down glycogen when there's already plenty of glucose in the circulation, such as after a meal. And you don't want to be making glycogen and storing glucose during fasting when the blood glucose is low. You need that glucose for energy. So one regulatory mechanism is that insulin inhibits glycogen phosphorylase and it stimulates glycogen synthase. So when you eat and the bloodstream is flooded with glucose, the pancreas releases insulin to allow that glucose to enter the cells. And at the same time, that insulin ends up inactivating glycogen phosphorylase. So you stop breaking down glycogen. And insulin stimulates glycogen synthase. So you start storing whatever extra glucose you have that you don't need right then. Now, on the other hand, imagine you're in a high stress state, a fight or flight state, where you need to share glucose. So what organ shares glucose? Primarily the liver. So your body communicates a high stress state by releasing epinephrine and glucagon. These hormones are known as counter-regulatory hormones because they do the opposite of insulin. And we've already seen that glucagon stimulates adenylyl cyclase in the hepatocytes to make cyclic AMP. That cyclic AMP activates protein kinase A. And we've seen how kinases can switch on certain enzymes by phosphorylating them. So protein kinase A activates glycogen phosphorylase kinase, and that in turn activates glycogen phosphorylase in a roundabout way. Don't get too hung up on the details. The bottom line is that glucagon stimulates glycogen phosphorylase and it stimulates glycogenolysis. That's how glucagon raises the blood sugar. Epinephrine does basically the same thing. Beta-adrenergic receptors are GS receptors, which also stimulate adenylyl cyclase. Remember that GS, the S is for stimulate. And that ends up activating glycogen phosphorylase and again stimulating glycogenolysis. When we discussed glycolysis, we talked about diseases that result from deficiencies of enzymes in the glycolytic pathway, such as pyruvate kinase deficiency. Well, there are also some diseases that result from deficiencies of enzymes of the glycogenolysis pathway. These are collectively called glycogen storage diseases. There are about 11 different glycogen storage diseases, but you only have to know 4 of them. But these 4 are very high yield. The first one I want to discuss is known as McArdle disease, which is a deficiency in glycogen phosphorylase. This is also called glycogen storage disease type 5. With McArdle disease, there's accumulation of excess glycogen in the muscle because all the enzymes that make glycogen are intact, but these patients can't break down glycogen. Specifically, they can't break down those alpha-1,4 linkages in glycogen. So the muscle cells fill up with glycogen, and the osmotic pressure from the glycogen draws water into the cells, and the muscle cells swell up and eventually lyse. And when muscle cells lyse, they spill myoglobin into the serum, and this is a condition called rhabdomyolysis. So that myoglobin goes through the blood to the kidneys, and that causes myoglobinuria, myoglobin in the urine. And the myoglobin can clog up the kidneys and eventually cause renal failure. Now, patients with McArdle disease can also have muscle cramping because their muscles are starved for glucose because they can't break down glycogen. But the good news is that patients with McArdle disease have a normal life expectancy. It's just a problem with the muscle cells. And again, it's because you're deficient in the enzyme glycogen phosphorylase. The next disease is von Gierke disease, or glycogen storage disease type 1. Now, this is a deficiency of glucose 6-phosphatase. And remember, this was the enzyme at the final step of both glycogenolysis and gluconeogenesis. Also remember that skeletal muscle cells don't have glucose 6-phosphatase. So this is a deficiency of glucose 6-phosphatase in those places that would share glucose, such as the liver, the enterocytes, and the kidney. So if these energy-sharing tissues can't perform either gluconeogenesis or glycogenolysis, that means they can't share glucose when the blood glucose starts to drop. So these patients are prone to hypoglycemia when they're fasting. So one characteristic of von Gierke disease is severe fasting hypoglycemia. These patients also have buildup of glycogen in the liver, because again, the liver can make glycogen, it just can't completely break it down. So this causes enlargement of the hepatocytes and hepatomegaly. These patients also get enlargement of the kidneys, they get elevated serum lactate, they get elevated uric acid, and hypertriglyceridemia. And then you can treat von Gierke disease by feeding the patient frequently so that they're going to have a fairly steady stream of dietary glucose to prevent that hypoglycemia. And then at night, you give them cornstarch to prevent overnight hypoglycemia because cornstarch is going to be digested more slowly. The next disease is Cori disease, or glycogen storage disease type 3. Cori disease is like a milder form of von Gierke disease. Cori disease is due to a defect in the debranching enzyme. So these patients can break down the alpha-1,4 linkages. They just can't break down the alpha-1,6 linkages. So glycogen can be broken down, just not completely. And gluconeogenesis works perfectly fine. So these patients have milder hypoglycemia. They have no elevation in lactate levels and no elevation in uric acid. And then the last disease is called Pompe disease, or glycogen storage disease type 2. Pompe disease is caused by a deficiency of the alpha-1,4 glucosidase in the lysosomes. So the lysosomes are responsible for breaking down a small amount of glycogen. So if the muscle cell lysosomes don't have this alpha-1,4 glucosidase, some of the glycogen won't get broken down. Now, there are two forms of Pompe disease. There's an infantile form and an adult form. The infantile form is more severe. These individuals have very, very low levels of enzyme activity, so symptoms appear earlier in infancy. The infantile form of Pompe disease causes severe muscle weakness, cardiomegaly, and heart failure, and these patients have a shortened life expectancy, usually less than a year if the disease is left untreated. Then the adult form of Pompe disease, or late onset Pompe disease, occurs when you have some lysosomal enzyme activity, but less than normal. So these patients generally have no cardiac involvement. They get gradual onset of skeletal muscle weakness, as well as diaphragm weakness and respiratory failure. Now it's time for the end of session quiz. So I want you to pause the video, work through those questions, then restart the video and we'll go over the answers. First question, what's the rate limiting enzyme for glycogen synthesis? It's glycogen synthase. And then for glycogenolysis, it's glycogen phosphorylase. So again, make sure you know all those rate limiting enzymes for all these pathways. Next, which enzyme converts glucose 6-phosphate to glucose? It's the enzyme glucose 6-phosphatase. Remember, that's the enzyme that's deficient in von Gierke disease. Last question, which glycogen storage disease matches each of the following phrases? So glycogen phosphorylase deficiency is McArdle disease. Glucose 6-phosphatase deficiency is von Gierke disease. We just mentioned that. Lactic acidosis, hyperlipidemia, and hyperuricemia is von Gierke disease. Alpha-1,6 glucosidase deficiency, that's debranching enzyme, which is deficient in Cori disease. Alpha-1,4 glucosidase deficiency is Pompe disease. Cardiomegaly is seen with Pompe disease, the infantile type. And then diaphragm weakness and respiratory failure is adult type Pompe disease. Increased glycogen in the liver and severe fasting hypoglycemia is von Gierke disease. Hepatomegaly, hypoglycemia, hyperlipidemia, but with normal kidneys, normal lactate, and normal uric acid is Cori disease. Painful muscle cramps and myoglobinuria with strenuous exercise is McArdle disease. And severe hepatosplenomegaly with enlarged kidneys is von Gierke disease. So if you know nothing else about these diseases, know those bullet points. Very high yield. And that's it for now. I'll see you next time.
It's time for the 3-question warm for Biochem 10. Let's get going. What is the landmark for a pudendal nerve block? So in this image, we're seeing the pudendal nerve block. You have to palpate for the ischial spine to direct the nerve block. So this is used for pain control during a vaginal delivery. Next, what enzyme catalyzes the rate-limiting step in carbohydrate digestion? So these are the oligosaccharide hydrolases, which are found at the intestinal brush border. Next, what vitamin deficiency results from Hartnup disease? So Hartnup is a deficiency of tryptophan, which leads to niacin deficiency. So don't forget that niacin is also vitamin B3. And niacin or B3 deficiency is also the name of pellagra. All right, so that's going to be it for the warm-up. Let's get to that lecture now.
In this video, we're going to discuss production of ATP. We've talked about glycolysis and how you convert glucose into pyruvate, but that really doesn't generate a ton of ATP. So we need to figure out how to feed that pyruvate into the TCA cycle and then the electron transport chain. There are actually 4 different things you can do with pyruvate. Pyruvate can be converted to oxaloacetate using the enzyme pyruvate carboxylase. We talked about that previously because that's the first step of the gluconeogenesis pathway. Pyruvate can be converted to acetyl-CoA using the enzyme pyruvate dehydrogenase, and that's going to be fed into the TCA cycle. Pyruvate can be converted to lactate, which occurs in anaerobic respiration. So cells that lack mitochondria, such as red cells, or cells that aren't getting enough oxygen for aerobic respiration, are going to put glucose through the glycolysis pathway to make pyruvate plus a little ATP, and then they convert the pyruvate into lactate in order to get rid of it. And that's going to go into the Cori cycle, which we'll talk about shortly. And then finally, pyruvate can be converted to the amino acid alanine, and we'll talk about the alanine cycle shortly as well. Let's talk about how we convert pyruvate to acetyl-CoA for the TCA cycle. The enzyme complex that converts pyruvate to acetyl-CoA is called pyruvate dehydrogenase, and this process is heavily regulated because there are so many different things you can do with pyruvate. If we're going to commit to converting pyruvate into acetyl-CoA so that it can go into the TCA cycle, you want to make sure that that's really what the body needs. Pyruvate dehydrogenase is regulated by the fact that it requires a lot of cofactors. And the mnemonic for these cofactors is TLC for nobody, tender loving care for nobody. The T is for thymine pyrophosphate, or TPP. That's the active form of thymine. That's vitamin B1. L is for lipoic acid. C is for coenzyme A, which is ultimately derived from vitamin B5. The F in 4 stands for FAD, which comes from vitamin B2. And the nobody stands for NAD+, which comes from vitamin B3, which is niacin. So several B vitamins are required to convert pyruvate to acetyl-CoA. Vitamin B1 to make TPP, B2 to make FAD, B3 to make NAD+, and B5 to make CoA. So make sure you know all those cofactors. You should also know that lipoic acid can be inhibited by arsenic, which manifests clinically as garlic breath. So if on your test you're given a patient with breath that smells like garlic, think about arsenic. These patients will have GI problems like vomiting and rice water stool as well. So arsenic poisoning is going to look like gastroenteritis plus garlic breath. Now, what if you have pyruvate dehydrogenase deficiency so that you can't make acetyl-CoA? Well, in pyruvate dehydrogenase deficiency, you get a backup of pyruvate, and that pyruvate is going to be converted to other things such as lactate, resulting in lactic acidosis. It can also back up and be converted to alanine. And then besides lactic acidosis, this will also result in neurological defects. Pyruvate dehydrogenase deficiency can either be an X-linked congenital defect where you're deficient in the enzyme, or it can be an acquired defect like from arsenic exposure. We just said that arsenic interferes with lipoic acid, so that would be an acquired pyruvate dehydrogenase deficiency. Or if you're deficient in one of those B vitamins needed to make the necessary cofactors, that could manifest as pyruvate dehydrogenase deficiency as well, especially vitamin B1 deficiency, thymine deficiency. Now, when do we classically see thymine deficiency? In alcoholics. So thymine deficiency may lead to a functional pyruvate dehydrogenase deficiency. And then how do we treat it? Well, if it's secondary to a vitamin deficiency, you're going to supplement those vitamins. But these patients also need to make acetyl-CoA from something other than glucose and pyruvate. Maybe they need to make acetyl-CoA from fats. So putting the patient on a high-fat diet can also be important. And you also want a diet that's high in ketogenic amino acids like lysine and leucine, because ketone bodies can also be used for energy instead of glucose. Assuming that your pyruvate dehydrogenase complex does work, you convert pyruvate into acetyl-CoA. Then what? Well, then you're going to feed that acetyl-CoA into the TCA cycle, which is also called the Krebs cycle or the citric acid cycle. So several different names for the same biochemical process. The good news is that for step 1, you do not need to memorize every step of the TCA cycle, but you do need to know 3 key enzymes. The first enzyme is citrate synthase, which adds acetyl-CoA to oxaloacetate to make citrate. The next one worth knowing is isocitrate dehydrogenase, which we said was the rate-limiting enzyme of this process. And then there's a highly regulated enzyme called alpha-ketoglutarate dehydrogenase. So make sure you know those three enzymes as TCA cycle enzymes: citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. And the reason that alpha-ketoglutarate dehydrogenase is a highly regulated step is that it requires the same cofactors as pyruvate dehydrogenase: TPP, lipoic acid, coenzyme A, FAD, and NAD+. So again, the mnemonic TLC for nobody applies to both pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase. I don't necessarily think it's important to memorize exactly what comes off the TCA cycle at each step. For every acetyl-CoA you put in, you get out 1 GTP plus some electron donors. You're going to get 3 NADHs and 1 FADH2. Plus you're liberating CO2, which has to go into the blood and then be exhaled by the lungs. But overall, these details aren't all that high yield for step 1. Now we're ready to use those electron donors to make some ATP. So let's look at the study guide and talk about the electron transport chain and oxidative phosphorylation. First of all, where does all this take place? Well, it takes place in the mitochondria, specifically on the inner mitochondrial membrane. So mitochondria have an outer mitochondrial membrane and an inner mitochondrial membrane, and this takes place on the inner membrane. So let's look at this process with the end in mind and then work our way backward. The whole purpose of the electron transport chain is to transfer hydrogen ions from the mitochondrial matrix into the intermembranous space between the two mitochondrial membranes. And if you can put hydrogen ions in there, you can then use that hydrogen gradient to power an ATPase that will generate ATP. So that's basically how the electron transport chain makes ATP, by moving hydrogen ions. But in order to move hydrogen ions into the intermembranous space, you have to do something with the electrons from that hydrogen. That's why it's called the electron transport chain. As you move along the chain from left to right, you're putting more and more hydrogen ions into the intermembranous space. Now the electron transport chain is a series of enzyme complexes. So there's complex 1, which is an NADH reductase. So that converts NADH into NAD+, plus an electron, and that gets transported to complex 2. So for every mole of NADH you put in, you can generate 2.5 moles of ATP. Then complex 2 is succinate dehydrogenase, which requires coenzyme Q. And complex 2 is where FADH2 is fed into the electron transport chain. So for every mole of FADH2 you put into the electron transport chain, you're going to get out 1.5 moles of ATP. Next is complex 3, which includes cytochrome B and cytochrome C1, and then that passes the electron to cytochrome C. And then complex 4 is cytochrome A and A3. And at the end of this complex, you donate the electron to oxygen and hydrogen ions to create water. But again, the important part of this whole process is that all along the chain, hydrogen ions are being pumped into the intermembrane space against their concentration gradient. And then eventually these hydrogen ions come back in, down the gradient, via ATPase, which uses that electrochemical gradient to generate ATP. So that's the electron transport chain. So what kinds of things interfere with this process? This is probably worth knowing. Complex 1 can be inhibited by amytal, which is a barbiturate, and also by rotenone, which is a piscicide, and also an insecticide. So a piscicide is a fish poison. It's used to eradicate invasive fish species or parasitic fish species. But rotenone inhibits complex 1. And another substance that inhibits complex 1 is MPP, which stands for methylphenylpyridinium. Now, sometimes when people go all Jesse Pinkman and try to cook up their own synthetic version of the opioid meperidine, they also cook up a byproduct called MPTP, and that MPTP gets metabolized to this MPP. So NPP inhibits complex 1 and can cause Parkinsonism. Then moving on to complex 3. Complex 3 can be inhibited by another fish poison called antimycin A. And then complex 4 is inhibited by a lot of things that can be abbreviated. So cyanide, which can be abbreviated CN, so cyanide can inhibit complex 4. Sodium azide, and azide is abbreviated N3, and that can inhibit it. Carbon monoxide, or CO, can inhibit it. And also hydrogen sulfide, or H2S. So CN, N3, CO, and H2S can all inhibit complex 4. And then finally, the ATPase enzyme can be inhibited by the macrolide antibiotic called oligomycin A. It's not an antibiotic we use clinically, but it's still a macrolide. Another way to disrupt the electron transport chain is to give something called an uncoupling agent. An uncoupling agent basically uncouples the electron transport chain from the ATPase. So it increases the permeability of the inner mitochondrial membrane to hydrogen ions. So if the hydrogen ions in the intermembranous space can leak back into the mitochondrial matrix down their concentration gradient without going through an ATPase, that potential energy is going to be wasted, and it's wasted as heat. So uncoupling agents make the inner mitochondrial membrane more permeable to hydrogen ions, and they generate more heat in the body. There's a substance called thermogenin, which is found in brown fat, that hibernating animals use to generate more heat in the winter. They don't need to generate ATP during hibernation. They really need that energy to be converted to heat so they stay warm while they're hibernating. So thermogenin in brown fat can act as an uncoupling agent, and so can high-dose aspirin. So in cases of aspirin overdose, as this uncoupling takes place, patients can become hyperthermic. And then a third uncoupling agent is called 2,4-dinitrophenol, or 2,4-DNP, which is used illegally for weight loss because it ramps up the metabolism and you burn all this energy as heat. And then you get hyperthermic and you die. So, not a good idea. Before we move on, let's do a quick review.
Welcome back to everybody's favorite game, Metabolism Mania. Our first question, what 4 end products can be made from pyruvate? Oxaloacetate, acetyl-CoA, lactate, and alanine. Great job! Next question, what 4 B vitamins are needed to make cofactors for pyruvate dehydrogenase? B1 or thiamine, which makes thiamine pyrophosphate or TPP. B2 or riboflavin, which makes flavin adenine dinucleotide or FAD. B3 or niacin, which makes NAD+. And B5 or pantothenic acid, which is used to make coenzyme A. And of course, the 5th required cofactor for pyruvate dehydrogenase is lipoic acid, which is the only one not derived from a B vitamin. And our final question, which TCA cycle enzyme also requires the same cofactors as pyruvate dehydrogenase? Alpha-ketoglutarate dehydrogenase. Great job! We'll be back to spin the big prize wheel right after this word from our sponsor.
Now let's go back and briefly discuss the other 2 potential fates of pyruvate. We talked about how you can convert pyruvate to oxaloacetate for gluconeogenesis. And we've just finished talking about converting pyruvate to acetyl-CoA for the TCA cycle. So now let's talk about lactate in the Cori cycle. On the left, we have good old-fashioned anaerobic glycolysis, where you break down glucose into 2 pyruvate molecules, and then you're netting 2 ATPs in that process. Remember that? 2 ATPs isn't really a great return when you consider you could be getting 30 or 32 ATPs for that glucose molecule. But this is either taking place in a red blood cell where you don't have any mitochondria, or maybe it's taking place in a muscle cell that it's used up all its available oxygen. So pyruvate molecules are converted into 2 lactates using the enzyme lactate dehydrogenase, or LDH. Then the cell needs to be able to get rid of all that lactate. So the lactate leaves the cell and goes into circulation and eventually makes its way to the liver. Then the liver will convert the lactate back to pyruvate, again using that same enzyme, LDH. And then pyruvate can be used for gluconeogenesis to generate some more glucose. And that glucose can be shared with all the cells that need it. But this conversion of 2 lactates back to glucose consumes 6 ATPs. So it uses a lot of energy relative to the 2 ATP that the first cell generated. So it's a good thing that the liver stores a lot of energy. The other thing about lactate or lactic acid is that when the cells are producing lactate because they're starved for oxygen, that lactate is a signal that tells the body, hey, I could use a little more oxygen down here. So lactic acid is a great communicator of oxygen starvation. And then the 4th fate of pyruvate is to convert it to the amino acid alanine. So let's look at the alanine cycle, which is similar to the Cori cycle. Just like the Cori cycle, with the alanine cycle you have the muscles making something that goes to the liver to be made into glucose. Except now you're using alanine instead of lactate. So pyruvate in the muscle can be converted to alanine, then excreted into the circulation and taken up by the liver. Then the liver converts it back to pyruvate, and pyruvate is made back into glucose via gluconeogenesis. And then the liver can share that glucose with the tissues that need it. But the real purpose of the alanine cycle is not to make more glucose, but rather to deal with nitrogen. So in your study guide it says, why are alanine and glutamine found in such high concentrations in the blood? Alanine and glutamine are the 2 major carriers of nitrogen from tissues. Nitrogen comes mainly from amino acid metabolism, and your body needs to be able to eliminate that nitrogen. Excess nitrogen is ultimately excreted as urea, which is made in the liver, We're going to cover that in another video on amino acid metabolism. But the liver uses glutamine to make urea. It pulls nitrogen off of glutamine and puts that nitrogen into the urea cycle, which is what you see in the diagram. And we're going to go over the urea cycle in another video. But for now, let's look at the upper left part of this pathway in the muscle cell. Muscle cells break down protein into raw amino acids, and then the nitrogen from the amino acids gets transferred to an alpha-ketoglutarate, which generates glutamate plus alpha-keto acids. And glutamate can also pass off that nitrogen to pyruvate to form alanine, which simultaneously regenerates the alpha-ketoglutarate. This transfer of nitrogen-containing amino groups is called transamination. So skip down to the study guide question that says, what enzymes catalyze transaminase reactions? The enzymes are called transaminases, which always makes me think of Smokey and the Bandit. They're also called aminotransferases. Two names for the same family of enzymes, transaminases or aminotransferases. Transaminases transfer the nitrogen-containing amino group from an amino acid to alpha-ketoglutarate to form glutamate. Then what's left after you deaminate an amino acid is a keto acid, like pyruvate. And keto acids can be used for energy metabolism. So we're breaking down proteins into amino acids and converting amino acids into keto acids, like pyruvate, which is an energy substrate. So if you have a muscle cell that needs to break down protein for energy, this is a process it can use. But then it has to ship the nitrogen over to the liver, put it through the urea cycle, and make urea, which can be excreted in the urine. Then the next question says, what are the two most important transaminases, and what reactions do they catalyze? Transaminases are named based on the amino acid that donates the amino group to alpha-ketoglutarate. So alanine transaminase, which is commonly abbreviated ALT, is going to transfer an amino group from alanine to alpha-ketoglutarate, and that makes pyruvate and glutamate. And then aspartate transaminase, or AST, transfers an amino group from aspartate to alpha-ketoglutarate, which gives you oxaloacetate and glutamate. So what makes these enzymes ALT and AST so important? Well, we use them clinically as markers of liver damage. When your hepatocytes are injured, AST and ALT will spill into the blood. That's one of the ways we know that liver cells have been damaged. And the next question says, what cofactor is required by all transaminases? Well, it's pyridoxal phosphate, which is a derivative of vitamin B6. So once again, the transaminases and the alanine cycle allow you to move nitrogen from the muscle cells to the liver so that the liver can convert the nitrogen to urea. And then urea goes to the kidneys and is excreted in the urine. Because if you're going to use amino acids for energy, it's very important to be able to get rid of the nitrogen. So now we're ready for the end of session quiz. So pause the video, work through those questions, and we'll go through the answers. All right, first question. What are the possible products of pyruvate? Well, it's oxaloacetate, acetyl-CoA, lactate, and alanine. Next, what are the two main nitrogen transporters in the blood? We mentioned alanine and glutamine. Next, what are the major regulatory enzymes of the citric acid cycle, or the TCA cycle? Well, there's citrate synthase, which is the first step, isocitrate dehydrogenase, which is the rate-limiting step, and then alpha-ketoglutarate dehydrogenase is the heavily regulated step, which requires all those cofactors. Next, what substances are known to inhibit the complexes of the electron transport chain? We mentioned amytal, rotenone, MPP, antimycin A, cyanide, azide, carbon monoxide, and hydrogen sulfide. And the last question, what substances can increase the permeability of the inner mitochondrial membrane, thereby decreasing ATP synthesis but increasing heat generation? We mentioned aspirin, thermogenin, and 2,4-dinitrophenol. That's it for now. I'll see you next time.
It's time for another warm-up review. Let's get going. For what conditions is dantrolene useful? Well, you can use dantrolene for malignant hyperthermia or for neuroleptic malignant syndrome from certain antipsychotic drugs. Next, what are the possible etiologies of acute pancreatitis? So the mnemonic is actually the word pancreatitis. P is for hyperparathyroidism because that can potentially cause hypercalcemia. Alcohol, neoplasm, cholelithiasis, a big one. RX is for the R, so that's drugs. ERCP can lead to this. Abdominal surgery. Hypertriglyceridemia is a T there. Infection, specifically mumps. Trauma. Idiopathic and then scorpion sting, the one that never really happens in real life, but we still like to test on it. Next, which cell type releases renin? So the JG cells, or the juxtaglomerular cells. So they secrete renin in response to decreased blood flow in the kidneys. All right, that's it for the review. Let's get to that video.
We've already talked a lot about glucose, and in this video we're going to talk about some other processes related to sugar metabolism, starting with the hexose monophosphate shunt, or the HMP shunt, also known as the pentose phosphate pathway. So the HMP shunt is essentially a mechanism for generating a very important molecule called NADPH, not NADH, but NADPH. So first let's talk about why NADPH is so important. NADPH is used for synthesizing fatty acids and cholesterol. You need NADPH to generate oxygen-free radicals to kill bacteria in your phagocytes. NADPH is also needed inside red blood cells to protect the red blood cells from oxygen-free radicals. So not only is NADPH used to generate oxygen-free radicals in the phagocytes, it's also used to reduce oxygen-free radicals in red blood cells to prevent damage. And then the fourth reason why you might need NADPH is for the cytochrome P450 enzymes. So 4 very important reasons why this is such an essential molecule. So how do we get NADPH? Well, we get it through this pentose phosphate pathway, this HMP shunt. But there's really only one key step in this pathway that you need to know. So here are the nuts and bolts. You start with glucose 6-phosphate. Remember, when you first bring glucose into the cell, you phosphorylate that glucose with either hexokinase or glucokinase in order to trap it in the cell. So you start with glucose 6-phosphate, and then there are several reactions that take place. The main enzyme you need to know in these reactions is glucose 6-phosphate dehydrogenase, or G6PD. And that enzyme converts glucose 6-phosphate to a sugar called ribulose 5-phosphate. And you also generate NADPH. And you can use that ribulose 5-phosphate to generate PRPP, which is essential for nucleotide synthesis. That provides the ribose backbone in ribonucleic acids. Now let's look at how phagocytes use NADPH to generate reactive oxygen species and free radicals, and then what those free radicals are used for. So this is known as the oxidative burst process, or it's called a respiratory burst. So this takes place inside the phagolysosome after the phagocyte has engulfed a bacterium or something else it wants to destroy. So in order to generate oxygen-free radicals, you need oxygen molecules, NADPH, and the enzyme NADPH oxidase, which catalyzes the reaction. You should know that enzyme for your test. Now once the phagocyte has made a reactive oxygen species, you can convert that to hydrogen peroxide. And the enzyme that catalyzes that process is called superoxide dismutase. And then a third enzyme, myeloperoxidase, can use hydrogen peroxide to generate hypochlorous acid. So those are 3 important enzymes for your phagolysosome: NADPH oxidase, superoxide dismutase, and myeloperoxidase. And these 3 enzymes make those caustic substances that destroy bacteria inside the phagolysosome. Now, back in immunology, we talked about an immunodeficiency disease that was caused by a deficiency of NADPH oxidase. Remember what that was? Chronic granulomatous disease. Because these patients don't have NADPH oxidase, these patients' macrophages can't produce those caustic substances needed to digest bacteria. But the macrophages can take hydrogen peroxide from the environment and use that to kill bacteria. But some organisms, like Staph aureus and also Aspergillus species, make an enzyme called catalase, and that turns hydrogen peroxide into water. So patients with chronic granulomatous disease are susceptible to infection by these catalase-positive organisms because these organisms can neutralize the environmental hydrogen peroxide before the phagocytes have a chance to use it against them. Now I said earlier that NADPH was used both to generate reactive oxygen species by the immune system, and it's also used by red cells to neutralize reactive oxygen species like hydrogen peroxide. So a red blood cell will take hydrogen peroxide and convert it into water using the enzyme glutathione peroxidase. Know that enzyme, glutathione peroxidase. It neutralizes hydrogen peroxide by letting it react with reduced glutathione, which we abbreviate GSH. So reduced glutathione is an important antioxidant, just like vitamin C and vitamin E and vitamin A. So you're going to see that as a recurring theme. Reduced glutathione is an antioxidant. So once reduced glutathione has reacted with hydrogen peroxide, it's no longer reduced glutathione. It's been oxidized. So now it's glutathione disulfide, which is GSSG. So now we need to convert that back to reduced glutathione. And the enzyme that does that is glutathione reductase. And that enzyme uses NADPH as an electron donor. Then to keep a steady supply of NADPH around, we need G6PD, glucose-6-phosphate dehydrogenase. All these enzymes are interconnected. So again, for red cells to detoxify hydrogen peroxide, they need reduced glutathione, and then to keep a steady supply of reduced glutathione, they need a steady supply of NADPH. And the enzyme that produces NADPH is G6PD. So what happens if you're deficient in G6PD? Well, you're not going to be able to make NADPH, and that kind of locks up this whole process. Because our red cells need NADPH so much to neutralize reactive oxygen species and to neutralize hydrogen peroxide that the red cells of patients with G6PD deficiency are going to be particularly susceptible to reactive oxygen species. So, when patients with G6PD deficiency take certain drugs, such as sulfa drugs, the sulfa drug can induce oxidative damage in the red blood cells, which the body can't repair because there's no NADPH. And that oxidative damage eventually leads to hemolytic anemia. And if you look at the red blood cells on the peripheral blood smear of a patient with G6PD deficiency, you're going to see Heinz bodies, which you can only see with a special stain, and also Bite cells. Heinz bodies are little clumps of oxidized hemoglobin that is precipitated within the red cells. And then Bite cells are red cells that look like somebody came along and took a big bite out of them. So as these abnormal red cells with Heinz bodies pass through the spleen, the macrophages of the spleen recognize them as abnormal and literally take a bite out of the cell to remove the Heinz bodies. So macrophages remove the Heinz bodies, and that results in Bite cells. In your study guide, there's a question that says, in patients with G6PD deficiency, ingestion of what substances can induce oxidative damage to red cells? So there are a bunch of drugs and one classic food that you need to know. And our mnemonic for these is spleen purges nasty inclusions from damaged cells. So the S is for sulfonamides, which we mentioned. P is for primaquine, which is an anti-malarial drug. N is for the antibiotic nitrofurantoin, which is rarely used for anything but UTIs. I is for the antibiotic isoniazid, or INH, which is used for TB. F is for fovabines, which is the only one on this list that isn't a drug, but it's a classic one worth knowing. D is for the antibiotic dapsone, which is mainly used for leprosy and sometimes gets used for PCP prophylaxis. And then the C is for chloroquine, which is another anti-malarial drug. And sometimes you'll see high-dose aspirin and NSAIDs on this list, and they're potentially a problem, but they're usually pretty safe at normal doses. And then one other thing worth knowing about G6PD deficiency is that it's an X-linked disorder, and it's hypothesized that G6PD deficiency may make red cells less susceptible to malaria. Now, there's another enzyme deficiency that we've already discussed that can also result in hemolytic anemia. That was a deficiency of one of the enzymes in the glycolysis pathway, such as pyruvate kinase deficiency. So, just to compare these two disorders, if you're deficient in a glycolytic enzyme like pyruvate kinase, the red cells can't make ATP, so they're going to lyse and they die that way. Now, in the case of G6PD deficiency, your cells can make ATP, but they can't protect themselves from oxidation. So in order for those red cells to survive, it needs to be able to both process energy and protect itself. Now we're ready to talk about some of the other sugars that your body can use for energy, other than glucose. So starting with fructose. Fructose is a simple sugar found in plants. I usually think of fructose as being found in fruit. Now, I don't want you to memorize the metabolic pathway for fructose. It's not really important for step one. But it should look fairly familiar because it shares some of the steps with the glycolysis pathway. And I do want you to know the two disorders that arise from this pathway. The first disorder is essential fructosuria, which occurs when you're deficient in fructokinase. Again, don't get bogged down in the pathway, but do remember that enzyme and the disorder that goes with it. Essential fructosuria, fructokinase. And because these patients can't break down fructose the normal way, that fructose is going to build up in the blood and then spill into the urine. Other than that, it's a benign disease. With some sugar disorders, the sugar is going to get trapped in the cell and water follows the sugar osmotically, and eventually the cell becomes swollen and you get osmotic damage, possibly even cell lysis. But with essential fructosuria, none of that happens. That's why it's benign. The only problem is that you can't utilize fructose as an energy source, so it's going to spill out into your blood and then into your urine. Another disorder of fructose metabolism is fructose intolerance, which is due to a deficiency of aldolase B. So with aldolase B deficiency, you're going to have an accumulation of fructose 1-phosphate, and you're essentially using up a lot of the phosphates because they're all stuck to fructose. So you're phosphorylating fructose, but then you can't do anything else with it. So you can't take that phosphate off. So that's why this is problematic. Now, as you use up phosphate, you end up inhibiting glycogenolysis and gluconeogenesis. which means you won't be able to correct fasting hypoglycemia. So as a result, with fructose intolerance, you're going to have hypoglycemia and vomiting, especially after consuming a lot of fructose, or sucrose, which is table sugar, because sucrose is just a disaccharide, a dimer sugar that's made up of glucose plus fructose. So these patients also have liver problems like hepatomegaly and jaundice. And we treat fructose intolerance by decreasing the intake of both fructose and sucrose. So again, know those two enzymes, fructokinase and aldolase B, and know which presentation results from a deficiency of which one. Now, one student suggested that deficiency of aldolase B causes the bad disease, so B for bad, because it causes hypoglycemia and vomiting and liver disease. And then you can remember that fructokinase deficiency is a kind disease. It's basically asymptomatic. Next, let's talk about galactose. So galactose is another monomer sugar that your body can use, like glucose or fructose. And deficiencies in the metabolism of galactose can also cause problems. Now again, don't memorize this pathway, but I do want you to know two disorders of galactose metabolism and the enzymes associated with them. The first is galactokinase deficiency. So galactokinase deficiency is due to a deficiency of galactokinase. Pretty straightforward. With galactokinase deficiency, you're going to have an accumulation of galactitol. So galactitol accumulates in the blood and it accumulates in the urine in the same way that fructose accumulates in essential fructosuria. Now, because galactose is accumulating, it can lead to cataracts, especially in infants. We call this infantile cataracts. So the lens of the eye is particularly sensitive to accumulation of sugars. We're going to see that with diabetes as well. But galactosidase deficiency isn't as bad as the next disorder, which is classic galactosemia. Classic galactosemia is due to the absence of the enzyme galactose-1-phosphate uridyl transferase. So galactose-1-phosphate uridyl transferase. And again, you're going to have excess galactose and excess galactitol in the blood and in the urine. You're going to have worse cataracts, plus hepatomegaly and jaundice, failure to thrive, and intellectual disability. Then the treatment is to exclude galactose from the diet, and also to exclude lactose, because lactose is a dimer sugar made of glucose and galactose. Now let's talk about lactose for a minute. Again, lactose is a dimer sugar, a disaccharide sugar that's found in milk. And the enzyme lactase breaks down lactose into glucose and galactose. So that enzyme lactase is found in the brush border of the small intestine. But if you have lactase deficiency, either because as you get older you just don't make enough lactase, or because you've some disease that transiently damages the brush border and that reduces lactase, but without lactase in the gut, you can't break down that dimer sugar. And your enterocytes can only absorb monomer sugars. They can't absorb dimer sugars. So the lactose remains in the lumen of the gut and it passes through the small intestine into the colon. Then the bacteria in the colon find that sugar and they just go nuts. They start consuming all that lactose. And as they consume all that lactose, they make a lot of gas like CO2 and methane. And all that gas causes bloating and cramping and flatulence. No laughing. And that lactose in the colon also draws water into the colon osmotically. And that's going to cause an osmotic diarrhea. So how do we treat this? Well, you can supplement lactase in your diet. Anytime a patient with lactase deficiency ingests some dairy, they can take a little Lactaid pill, and that has lactase in it. That's going to help them break down the lactose. Or they can just avoid dairy products altogether. All right, that's it for sugars. Let's go ahead and tackle the end of session quiz. First question, what's the rate-limiting step of the pentose phosphate pathway? It's glucose 6-phosphate dehydrogenase, or G6PD. Next, which tissues of the body use the pentose phosphate pathway? So remember, NADPH generated from this pathway is essential for reducing reactive oxygen species, and it's also used in the generation of cholesterol and fatty acids. So this pathway is especially important in red blood cells, in the liver, in the adrenal cortex, and the mammary glands during lactation. Next, explain why a deficiency of the enzyme that's the rate limiter for the HMP shunt can result in hemolytic anemia. So again, that rate-limiting enzyme is G6PD, and that's responsible for generating NADPH. So NADPH is used to produce reduced glutathione, and that's an important antioxidant. So without G6PD, RBCs are going to be more susceptible to oxidative damage, and that leads to hemolysis. Next, what are the symptoms of classic galactosemia? So you can see failure to thrive, intellectual disability, hepatomegaly and jaundice, and infantile cataracts. And the last question, what disorder is caused by a deficiency of each of the following enzymes? So deficiency of galactokinase causes galactokinase deficiency, which is that milder form of galactosemia. Aldolase B deficiency causes fructose intolerance. Lactase deficiency causes lactose intolerance. Galactose 1-phosphate uridyl transferase deficiency causes galactosemia. And the last one is fructokinase deficiency, which causes essential fructosuria. That's it for the HMP shunt. The Humpty Dance is your chance to do the shunt. All right, stop what you're doing, because I'm about to ruin the image and the style that you're used to.
These two guys don't get along, but when it comes to education, they are never wrong. Yeah, if you pay attention, you might get an A. They don't like each other, but that's okay, 'cause they are bosom buddies.
Hello everyone and welcome to the show. I'm Dr. Lewis and unfortunately again, this is Dr. McGinnis.
Hello.
How you feeling?
I feel fine.
You look a little pale, your eyes are, I don't know, a little cross-eyed, you're drooling.
Wait, hold on. I don't look anything like that. Why does everything out of your mouth have to be complete nonsense?
So wait, you're telling me that you don't look like that?
No, not even close.
That's weird because, every time I think of you, and again, I try not to think about you, the only picture I conjure up is a drooling, cross-eyed albino. Weird. Maybe it's your personality I'm thinking of.
You're such a drama queen. I got a question for you. You think your little brain can think about something other than video games and Cheetos for a few seconds?
We have Cheetos?
Focus, simpleton. What is G6PD deficiency?
You mean other than 3 letters, a number, and the word deficiency?
Yeah.
Well, G6PD stands for glucose-6-phosphate dehydrogenase, which is an enzyme involved in the pentose phosphate pathway. And it tends to be very important in maintaining a normal level of glutathione in red blood cells. Now, glutathione is needed to protect RBCs from oxidative damage from free radicals. Now, the deficiency is an X-linked recessive hereditary disease. So patients with it, when stressed by infection or certain drugs like the antimalarials and sulfonamides, or even by certain foods like fava beans, will have non-immune hemolytic anemia. How's that?
Barely adequate. But it was better than your usual slack-jawed mumbling.
Another gem of a lecture by good old Dr. Lewis. That's all for today. See you next time.
Bye.
It's time for the 3-question one for Biochem 12. Let's get going. What is the typical presentation of a patient with pancreatic insufficiency? So you might see diarrhea, steatorrhea, malabsorption and weight loss, and deficiency of the fat-soluble vitamins, so A, D, E, and K. Next, what are the symptoms of serotonin syndrome? So you're going to see muscle rigidity, hyperthermia, and cardiovascular collapse due to autonomic instability. Next, what artery prevents a horseshoe kidney from ascending in the abdomen? So this is your inferior mesenteric artery. All right, that's it for the warm-up. Let's get to that lecture now.
In this video, we're going to talk about starvation and malnutrition, or how our bodies generate energy when energy substrates are hard to come by. But first, since it's Take Your Child to Work Day here at DIT, I'd like to introduce my oldest son, Andrew.
Dad, is this where you make your videos?
This is it. See, we got the camera, we got the lights.
Awesome. Hey Dad, is there any way I could get an autograph?
Well, sure, I guess. I mean, it's kind of weird. I live with you. Why do you need my autograph?
No, I don't want your autograph. I want Dr. Lewis's autograph.
Let's talk about starvation, starting with how we transition from the fed state to the fasting state to a state of starvation. So what happens when you haven't eaten in several hours and your body doesn't have enough glucose remaining from the last meal? Well, the liver is the primary regulator of serum glucose levels. When you fast and blood glucose starts to drop, the liver will start making new glucose through gluconeogenesis. And it'll start mobilizing its glycogen stores, which is glycogenolysis. And then as you get farther and farther from your last meal and your glycogen stores become depleted, you're going to start making ketone bodies, which is a process known as ketogenesis. So let's go through some questions in the study guide. Look at number 4. It says, what fuels are produced and used in the post-absorptive period? So at this point, there are really just 2 fuels being produced. There's glucose from hepatic glycogenolysis and from gluconeogenesis, and there's fatty acids from adipose tissue. So fatty acids are stored in adipose tissue, and they represent another form of energy storage in your body beyond what's stored in the liver. And in the fed state, your adipocytes are happy. They're storing lots of energy. Then when you get to an energy deficient state, those adipocytes have to give up that energy again. So again, what's produced in the post-absorptive period? Glucose from hepatic glycogenolysis and gluconeogenesis, and then also fatty acids from adipose tissue. Then the fuel that's used is predominantly glucose. That's the go-to fuel for muscles and brain and other tissues. Your liver is able to generate a lot of glucose, so even though there may be some fatty acids being made, it's predominantly glucose that's being used. Then number 5 says, when does gluconeogenesis begin in the post-absorptive period, and when does it become fully active? So it begins 4 to 6 hours after the last meal. That's what's meant by the post-absorptive period, 4 to 6 hours after the last meal. And then gluconeogenesis becomes fully active when the glycogen stores are depleted, which is 10 to 18 hours after the last meal. So it takes at least 10 hours in a well-fed person with a healthy liver to deplete the glycogen stores. So if you're trying to enter ketosis, it's going to take a while. Now number 6 says, how does the pattern of fuel production and utilization change in early starvation? So we're beyond just an overnight fast now. We're not just in between meals. We're talking 24 hours after the last meal. So what's produced is glucose again, mainly from gluconeogenesis. There's not really much glycogenolysis going on anymore because you've used up most of your glycogen between 10 and 18 hours. So it's primarily glucose from gluconeogenesis. And then again, there's some fatty acids being produced from adipose tissue. And then what fuels your body uses depends on the tissue in question. So the brain uses predominantly glucose. The brain is very specialized. It really prefers glucose more than anything else, although it can use ketone bodies, but it really does need some glucose. But then muscles and other tissues use glucose sometimes, but they predominantly use fatty acids. So the rest of the body is going to use a secondary energy source like fatty acids much more readily than the brain will. The brain, again, primarily needs glucose, but muscles and other tissues can use fatty acids when needed. Then number 7 says in intermediate starvation, so this is 48 hours after the last meal, how does the pattern of fuel production and utilization change? So now we're 2 days since the last meal. For fuel production, you still have glucose from gluconeogenesis. Your liver can still maintain that. You still have fatty acids from adipose tissue. Now you're calling on those fatty acid reserves a lot more. So in intermediate starvation, your adipocytes are becoming much more important. And then you also have ketone bodies from the liver. And then what fuels are used? So again, the brain uses predominantly glucose, but also some ketone bodies. And then the muscles and other tissues use predominantly fatty acids, but also ketone bodies. Now, we're dealing with 3 main energy substrates. We talked about glucose, fatty acids, and now ketone bodies. And again, that process of generating ketone bodies is called ketogenesis. So in the liver, fatty acids and amino acids are metabolized into acetoacetate, which is a ketone body. And then you can use NADH to turn acetoacetate into another ketone body called beta-hydroxybutyrate. So you need to be able to recognize those 2 substances as the main ketone bodies, acetoacetate and beta-hydroxybutyrate. And lots of different tissues in your body, like muscle and brain, can use those ketone bodies for energy when there's not enough glucose around. You should also know that the rate-limiting enzyme for ketone body synthesis is HMG-CoA synthase, not HMG-CoA reductase. That's the enzyme that's inhibited by statins. We're talking about HMG-CoA synthase here. And not only can acetoacetate be converted into beta-hydroxybutyrate using NADH, but acetoacetate can also spontaneously become acetone. So as acetoacetate becomes acetone, patients that are making ketone bodies are going to start to have a slightly fruity smell to their breath from all this acetone. So that's one of the things you might notice in a patient that's in diabetic ketoacidosis, that fruity smell. Then another important point to know is that when you test for ketones in the urine of a DKA patient, you're actually only testing for acetoacetate. The urine test for ketones does not test for beta-hydroxybutyrate. So when would you start generating ketone bodies? Well, it's basically whenever you break down so many fatty acids and make so much acetyl-CoA that the TCA cycle can't handle it all. So you're going to start making ketone bodies. Now, think about people who go on an extreme no-carb diet like the Atkins diet. They want to start burning fat. They want to start breaking down fatty acids. They're trying to become ketogenic. So whenever there's prolonged starvation or a very, very low-carb diet, you're going to use up all your oxaloacetate because of gluconeogenesis. So using up all your oxaloacetate, you're depleting oxaloacetate. And as you deplete oxaloacetate, the TCA cycle has to slow down. So you're redirecting TCA cycle metabolites toward gluconeogenesis. So in times of excess gluconeogenesis, you're going to actually shut down the TCA cycle. When you shut down the TCA cycle, you're going to start making ketone bodies. So that brings us to number 8 in the study guide. What metabolic scenario favors the synthesis of ketone bodies? It's when the production of acetyl-CoA from beta-oxidation of fatty acids exceeds the oxidative capacity of the TCA cycle. And then number 9 says, true or false, ketone bodies can be used by all body tissues, including the brain. This is false. The brain can use ketone bodies, but red blood cells can only use glucose. So although the neurons of the brain prefer glucose, they can use ketone bodies, but red cells can only use glucose. Then, going back to our stepwise discussion of starvation, number 10 says, what's the pattern of fuel production and utilization in prolonged starvation? So not between meals, not overnight, not 48 hours. Now we're talking 5 days after the last meal. So what's being produced? Well, really, it's the same 3 things. You're still making glucose from hepatic gluconeogenesis. You're making fatty acids from adipose tissue, and then also ketone bodies. But what's different here is the pattern of fuel use. So at 5 days, the brain is predominantly using ketone bodies at this point. So the brain has finally given in. It's not using glucose anymore. It's primarily using ketone bodies. Then the muscle and other tissues are predominantly using fatty acids, but also some ketone bodies, and some of these tissues are going to need some glucose like the red blood cells. So predominantly fatty acids, but also ketone bodies and a little bit of glucose. And then number 11 says, comparing an overnight fast to a 3-day fast, what percentage of energy comes from glucose and from ketone bodies? So after an overnight fast, 90% comes from glucose, primarily from mobilizing glycogen stores. And only about 5% comes from ketone bodies. The other 5% or so comes from fatty acids. And then after a 3-day fast, 60% comes from ketone bodies and only 40% comes from glucose, primarily from gluconeogenesis, again, because you've already burned through your glycogen stores. Let's also talk a little bit about ethanol, because ethanol also can be used as an energy substrate. So we looked at this pathway for the metabolism of ethanol back in the farm video on drug metabolism. We talked about the 2 enzymes that break down ethanol. You have alcohol dehydrogenase that converts ethanol to acetaldehyde, and then acetaldehyde dehydrogenase converts acetaldehyde to acetate. And that acetate can ultimately be used to make acetyl-CoA. We're going to come back and talk about why acetyl-CoA is such an important molecule in just a second. But the thing I want you to focus on here is the fact that both of these reactions generate NADH. So picture this, if you will. You knock back a couple of drinks on an empty stomach. Since you're in that post-absorptive period, at least 6 hours after your last meal, your liver's supposed to be putting up a steady supply of glucose back into the circulation through gluconeogenesis, right? Well, when ethanol's around, the hepatocyte is going to preferentially metabolize the ethanol in order to clear it out of the system. So if the liver has to choose between gluconeogenesis and metabolizing ethanol, it's going to actually metabolize the ethanol first. So while the hepatocytes are busy metabolizing ethanol, they're making a lot of NADH. And in order to keep metabolizing ethanol, they've got to convert that NADH back to NAD. And then the way they do that, the way the liver does that, is by converting pyruvate to lactate and also by converting oxaloacetate to malate. So if there's no pyruvate and no oxaloacetate available, because it's all been converted to lactate and to malate to regenerate NAD, how on earth is gluconeogenesis supposed to take place? Well, it doesn't. So the bottom line is when your liver is metabolizing ethanol, it can't perform gluconeogenesis. And that can cause a severe fasting hypoglycemia in patients that consume a lot of alcohol in a fasting state. Now, I mentioned in passing that acetyl-CoA is a really important molecule. So we've talked about how pyruvate is converted into acetyl-CoA. We just talked about how alcohol can be converted into acetyl-CoA. So what the heck did your body do with all this acetyl-CoA? Well, there are lots of different things your body can do with it. First of all, we've already talked about how acetyl-CoA can enter the TCA cycle. That's what we typically think about with acetyl-CoA. Acetyl-CoA is also the starting point for fatty acid synthesis, and for cholesterol synthesis, and for ketone synthesis. Let's shift gears now and talk about malnutrition. There are 2 states of malnutrition you should be familiar with. The first one is kwashiorkor. So kwashiorkor is protein malnutrition. You may still be getting plenty of calories, but you're not getting enough protein. So with protein malnutrition, you start to get fatty liver disease. So one of the liver's primary jobs is making all sorts of protein. But if you have protein deficiency, there's no amino acid building blocks, so the liver can't make proteins. And it also can't make ApoB100. Now, ApoB100 is the apolipoprotein that's found on the lipoprotein particles that contain triglycerides and cholesterol. And if you can't make ApoB100 to put on those particles can't leave the hepatocyte. So those lipids just accumulate in the liver, and you get fatty liver disease. You're also going to have some edema with kwashiorkor because without adequate serum proteins, plasma is going to tend to leak out of the vasculature and get into the interstitium and cause edema. These patients can get anemia because they can't generate the proteins needed to make rapidly dividing cells like blood cells. These patients get skin lesions, and they also get depigmentation of the skin and of the hair. So the skin is constantly sloughing off dead keratinocytes and generating new ones. So you need a lot of protein to maintain this constant regeneration. So without adequate protein, you're going to be prone to getting skin lesions. So one mnemonic for the main features of kwashiorkor is the word flames. So the FL is for fatty liver, the A is for anemia, the M is for malnutrition, the E is for edema, and then the S is for skin lesions. Now this is a picture from the CDC. And you see this classic image of a malnourished, skinny child with muscle wasting, and they have a large, protuberant belly from the edema and ascites and hepatomegaly. This child also has thinning hair and some depigmentation of the hair and edema in his feet. Then the other malnutritional state is marasmus. Marasmus is a total energy malnutrition. So kwashiorkor is a protein malnutrition, but marasmus is a total energy malnutrition. It's not just protein that's missing. It's protein, it's fat, carbohydrates. Everything is deficient in the diet of a patient with marasmus. So marasmus results in muscle wasting, subcutaneous fat loss. Now, what if you have a patient with marasmus that suddenly starts eating again? Maybe a patient that's been on a 10-day fast and starts to eat again, or he was stranded on a desert island like Castaway or Gilligan's Island or something for an entire week. And then he's rescued and he can start eating again. What might happen in this scenario is something called refeeding syndrome. Refeeding syndrome can take place in a person who's had negligible nutrient intake for at least 5 days. So during a fasting state, your body has to maintain osmotic balance in the blood. This means that the cells have to unload things like potassium and phosphates into the blood. They're sacrificing their electrolytes and things into the blood. And basically that results in the cells becoming intracellularly depleted. And then what do we say was the primary energy substrate during a prolonged fast or a period of prolonged starvation? It's the conversion of fat to ketones. There's a little bit of gluconeogenesis going on, but your body's trying to conserve that protein and muscle tissue as best as it can. So it's going to sacrifice the fat tissue before it sacrifices the protein tissue as much as possible. Then when the patient is suddenly able to eat, and all of a sudden there's glucose and fats and protein available, all the cells start to say, hooray, we've got nutrients. And the body starts taking up all these nutrients from the blood and taking up potassium and magnesium and phosphate, all those things that they've been donating and they've become deficient in. Now they're able to scoop them all up. So in refeeding syndrome, as the cells start to pull nutrients from the blood, you get a drop in serum levels of magnesium and phosphate and potassium in particular. And that drop of magnesium and phosphate and potassium can cause arrhythmias and it can cause neurological problems. So when you feed someone after a prolonged fast, you need to kind of start low and go slow and monitor their electrolytes very, very closely. The other thing is that during this process of refeeding, there's going to be an overall depletion of ATP as cells phosphorylate things in order to trap them inside the cells. So as all those cells start to bring in glucose, they're going to phosphorylate the glucose, and that uses up ATP. So the cells may end up with an overall depletion of ATP, and that's a problem. All right, let's go ahead and complete the end of session quiz, and then we'll go over the answers. Let's go through these. First question, what's the primary energy source in a patient that has not eaten in 2 days? It's fatty acids. Next, what's the rate-limiting enzyme in ketone body synthesis? It's HMG-CoA synthase. Next, a stressed physician comes home from work, consumes 7 or 8 shots of tequila in rapid succession before dinner, and then becomes hypoglycemic. So the question is, why does she become hypoglycemic? So remember this mechanism. You generate a lot of NADH when you metabolize ethanol. And that NADH shunts pyruvate toward the production of lactate, and it shunts oxaloacetate toward the production of malate. And this means that pyruvate and oxaloacetate are no longer available for gluconeogenesis. So the patient's more likely to become hypoglycemic. And the last question, what are some of the hallmark features of kwashiorkor? So remember our mnemonic flames for fatty liver, anemia, malnutrition, specifically protein malnutrition, edema, and then the S is for skin lesions. That's it for now. I'll see you next time. Let's get warmed up with a review quiz. First question, what are the classic manifestations of Guillain-Barré syndrome? So Guillain-Barré syndrome causes symmetric ascending muscle weakness, so it starts in the lower extremities and then it ascends. There can be some facial paralysis, and you can also see autonomic dysfunction such as hypertension or hypotension. Next, what are the risk factors for the development of hepatocellular carcinoma? The risk factors include hepatitis B and C, hemochromatosis, alpha-1 antitrypsin deficiency, hepatic adenoma, and cirrhosis, basically cirrhosis from any cause. The last question, what's the presentation of tricyclic antidepressant overdose? The main findings are cardiotoxicity like arrhythmias and hypotension, CNS toxicity like coma or seizures, and anticholinergic symptoms. Those are the tri-Cs. Now let's get started with the lecture.
Hello everyone, welcome to our Step 1 video on Biochem 13, lipids. 1 second. Here's to my friend Andrew. Here's hoping you were adopted. Your hero, Dr. Lewis. All right. So now let's talk about lipid absorption, lipid processing, and lipid transportation. All about lipids today. So I want to briefly introduce some of the key players and their functions. So first, lipids are insoluble in water. Therefore, this poses a transport problem for the body. How are we going to get lipids to where they need to go? Now, to solve this problem, the body packages fats into things called lipoproteins. Now, as the name implies, lipoproteins are made up of lipids and proteins. Now, the protein actually acts to emulsify the lipids, so those lipids can be transported around in bodies. So, when we talk about lipids and lipoproteins, this includes a lot of things. Things like triglycerides, things like phospholipids, things like LDL, HDL. Now remember that LDL, the low density lipoprotein, which we all know is your bad cholesterol. And really it isn't really bad, it's just too bad if you have too much of it. Now LDL transports cholesterol from the liver to the tissues. HDL, your good cholesterol on the other hand, is going to transport cholesterol from the periphery back to the liver. And a good way to remember these two is that L in LDL you want to keep low, and the H in HDL is what you want to keep high. Now what about some other things? Things like chylomicrons, VLDL, and IDL. Well, chylomicrons deliver dietary triglycerides to peripheral tissues. They also deliver cholesterol to the liver in the form of chylomicron remnants. Now, VLDL delivers triglycerides to peripheral tissues, and it's going to be secreted by the liver. And finally, we have IDL, that intermediate density lipoprotein. And IDL is a byproduct that is formed in the degradation of VLDL. Now, IDL delivers triglycerides and cholesterol to the liver, and it's really important knowing these key players and their functions as we move forward. All right, so now let's get down into this. Let's look at this diagram of lipid transport. And yes, it is a little bit complicated. It might take us a little bit to master this. Now, here we see an exogenous pathway, which indicates lipids are originating in the diet. And then there's an endogenous pathway, indicating lipids that are produced or packaged in the body itself. So first, let's start here. Let's say we take in some dietary fat. We've eaten something, which is then broken down into small particles by pancreatic lipase. Now, these particles are absorbed by your enterocytes in the intestine. And those enterocytes take those and then form something called chylomicrons. Now, chylomicrons, as we mentioned before, they're about 90% triglyceride, they're about 5 to 10% phospholipid, and about 1 to 3% cholesterol. So a whole lot of triglyceride here. Now the next step is for those chylomicrons to go into the lymphatic system of the intestines. Now the only way that enterocytes can push out or package lipid out of into the lymph fluid is with the help of an apolipoprotein called ApoB48. If you don't have enough ApoB48, you can't push or package that chylomicron out, which then results in a disorder called A-beta lipoproteinemia, which literally means absence of beta lipoprotein. Now we'll talk about apolipoproteins and pathologies later, but keep that in mind. So let's get back to our story. Chylomicrons, they're released into the lymphatic system of the intestines, and then they travel through the thoracic duct and then get secreted into the bloodstreams at the level of the left subclavian vein. So now we have chylomicrons in the circulation. And as a chylomicron goes by an adipose cell or maybe a muscle cell or other types of cells, that cell might use something called lipoprotein lipase. Lipoprotein lipase, important to remember that. And that's to draw free fatty acids off of the chylomicrons. And this chylomicron particle, which was once big and swollen with triglycerides and fatty acids, is now much smaller because some of those fatty acids have now been removed. So what you end up with is a chylomicron remnant. Now, that chylomicron remnant then is going to travel to the liver. So what does the liver do to it? Well, the liver will take the chylomicron remnant and then synthesize something called a VLDL particle. Now, this VLDL particle can then be secreted by the liver into the circulation. And this is with the help of something called ApoB100. Now, if you lack ApoB100, then those particles are not going to leave the liver. So ApoB100 attaches to that VLDL and then leaves the hepatocyte and then goes into circulation. Remember, ApoB100, we'll come back to that. Now, as that VLDL goes into the circulation, it is still a very triglyceride-rich particle. It's going to go travel through the circulation. Again, it's going to pass by a bunch of cells, adipose cells, muscle cells, all sorts of cells. And just like with the chylomicron, the cells are going to use lipoprotein lipase on that VLDL, and it's going to pull off more fatty acids. And as a result, it's going to become a smaller particle again. It's going to become an IDL, so that intermediate density lipoprotein. So that IDL can then go throughout the circulation some more. Now, two primary pathways exist for that IDL. And now it can be reabsorbed by the liver, or it can be hydrolyzed further to produce an LDL particle. And this is through the actions of hepatic lipase, sometimes referred to as hepatic triglyceride lipase. And LDL is very cholesterol rich, so you pulled off nearly all the free fatty acids from that particle. So it's a very cholesterol rich particle. So why is LDL considered bad? Well, if it becomes oxidized, it actually causes problems in the arteries, such as the formation of an atheroma, which is that initial step of atherosclerosis. So that's why that LDL is bad. So what else can LDL do though? Well, LDL can be reabsorbed back into the liver if the liver feels like it needs some more cholesterol, but the cells of the body may also need LDL particles. It does serve a function. And so the cells of the liver and the cells of all the other types of cells are going to absorb these LDL particles by way of endocytosis, which is mediated by the LDL receptor. Now that's a very important receptor, and we'll come back to that when we talk about pathology. Now let's talk about the HDL particle. So HDL is produced by the liver and contains the ApoA1 apolipoprotein. Now recall that our other particles had ApoBs like ApoB48 and ApoB100. HDL is a very small particle. It contains more protein and very little triglyceride. The purpose of HDL is to scoop up cholesterol from the periphery and then return it back to the liver, primarily. Now, when the HDL is immature and very new, it contains very little lipid, and we call this the nascent HDL particle. Now, that nascent HDL particle takes up cholesterol from the periphery by way of something called LCAT. Now, LCAT stands for lecithin cholesterol acyl transferase, so LCAT. Now, because of its now higher lipid content, it's referred to as an HDL2 or HDL3. That's depending on its cholesterol content. So it's scooping up all this cholesterol, it's getting fatter, it's collecting all this stuff. Now, what's interesting, actually, HDL2 actually has a higher concentration of cholesterol in it. Now, another job of HDL is to exchange lipids with other lipoproteins, things like VLDL, IDL, and LDL. Now, the HDL will give these other particles cholesterol esters in exchange for triglycerides. And the enzyme that catalyzes the exchange is the CETP, or cholesterol ester transfer protein. So CETP transfers cholesterol off of the HDL onto either LDL or LDL particles. So now the HDL is full of triglycerides, it's fat. No, it's full. It now has to actually offload some of that lipid cargo so it can then go back out and scoop more stuff up. So first, what can it do? It can be hydrolyzed by hepatic lipase. Remember that we saw earlier the same thing, hepatic lipase working on our IDL particles. Well, this will pull off some of that triglyceride. Now again, that HDL is ready to go gather up more cholesterol in the peripheral tissues. Now the other method to get cholesterol off of an HDL particle is to travel back to the liver, where a receptor named scavenger receptor B1 or SRB1 can help actually pull cholesterol off of off those HDL particles at the level of the liver. All right, so we just mentioned several of the important apolipoproteins. Remember that an apolipoprotein is a protein that binds to lipids in order to transport it through the lymphatic or the circulatory systems. Now let's go through some of these in more detail. So ApoB48, remember that one. Remember, this is primarily involved in chylomicron secretion out of the enterocyte into the lymphatic system, ApoB48. Let's move on to ApoB100. Where was that one? Remember, that was found on that VLDL. Sometimes y'all see it on IDLs and LDLs as well. And this is what you need to have in order to leave the liver. So that ApoB100 attaches to that VLDL, and then you end up going into the circulation. Next, we have ApoE. So ApoE is going to mediate extra remnant uptake. So E for the apolipoprotein E, and then E for extra remnant uptake. Next, we have ApoA1. Remember, this is going to activate LCAT, and this is found on HDL particles. Remember, HDL particles, ApoAs instead of Bs. So A1 activates LCAT. And remember from our diagram that LCAT was used to move cholesterol from the cells into that HDL to help scoop up that extra cholesterol. And then we have a new one, ApoC2. So ApoC2 is a cofactor, C2 cofactor, for lipoprotein lipase. So C2 cofactor, and that's about it for our apolipoproteins. Extremely high yield, very high yield, worth knowing for your exam. So next, let's talk about some pathology, and this is first related to the apolipoproteins. So first, A-beta lipoproteinemia, long word. So this is a rare autosomal recessive disorder, and it's going to involve the deficiency of ApoB48 and ApoB100. So again, that ApoB48 used for chylomicron secretion to get it into the lymphatic system. So without ApoB48, enterocytes are never getting rid of their chylomicrons. Now, it's caused by a mutation in a gene for something called microsomal transfer protein, or the MTP gene. Now, this is needed for the production of ApoB48 and 100. Now, as a result, decreased chylomicron and VLDL synthesis and secretion occurs. Now, it presents in the first few months of life with steatorrhea, and this will make sense in just a minute. So how does this work? Well, without that ApoB48, enterocytes can't package and they can't get rid of those chylomicrons. So they build up in the cell, and then they can't absorb any further lipids from the intestines, so they stay there. Therefore, all that fat is going to stay in the intestines. Extra fat in the stool causes that nasty, nasty steatorrhea. Also important, these patients will have impaired transport of the fat-soluble vitamins. Which ones are those? Remember, it's ADEK, A, D, E, and K. Now, on intestinal biopsy, you're going to see enterocytes that are swollen with triglycerides. You're also going to see something called acanthocytosis of red blood cells on the peripheral smear. Now, this is where the plasma membrane of the red blood cell becomes kind of misshapen, kind of spiky in appearance. And this is due to an alteration in the membrane lipids. So these patients can also have ataxia and night blindness. So that ataxia is mainly from a vitamin E deficiency. The night blindness is more from vitamin A. Now, the treatment will include vitamin E primarily. Vitamin E helps to restore these lipoproteins. So remember, when you see ataxia, night blindness, some acanthocytosis, we're talking about A-beta lipoproteinemia. And then remember again, vitamin E is going to be your primary treatment for this. Now let's move on to some dyslipidemias and some more pathologic lipid problems. And there are really only 3 dyslipidemias that I want you to know about. The first one is hyperchylomicronemia, now which is an autosomal recessive condition. Now here we see a deficiency in lipoprotein lipase or a defective ApoC2. Remember that ApoC2 was a cofactor for lipoprotein lipase. So what you'll find in the blood are increased levels of chylomicrons, triglycerides, and cholesterol. And these patients will present with pancreatitis, and that's mainly due to an increased triglyceride. You're also going to see things like hepatosplenomegaly, maybe some pruritic xanthomas, as from the increased cholesterol, and we'll discuss that in just a minute. Xanthomas as well. Now, one last thing you need to know about the type 1 hyperchylomicronemia is that there is no increased risk for atherosclerosis, which actually kind of seems a little counterintuitive. Now, let's move on to type 2A, familial hypercholesterolemia. Now, this is an autosomal dominant condition with absent or decrease in LDL receptors. I remember we'd come back to this one. So LDL receptors are necessary to facilitate the uptake of circulating LDL into cells. Now, as a result, you're going to get elevated levels of LDL in the blood. Now, these patients will present with tendinous xanthomas, notably on the Achilles tendon. You're going to see the corneal arcus as well. And finally, an important note here is that this condition causes accelerated atherosclerosis. So individuals with a double dominant mutations can even have heart attacks as early in their 20s. So if you see a young, otherwise healthy person presenting with an acute MI, I want you to immediately think about a type 2A familial hypercholesterolemia. And the third dyslipidemia is the type 4 hypertriglyceridemia. So this is an autosomal dominant condition again, and that's going to lead to an overproduction of the VLDL in the liver. So you'll see an increased level of VLDL and triglycerides. And I want you to remember again, if you have really high levels of triglycerides, one of the main problems is pancreatitis. All right, that's it for dyslipidemias. So let's talk about some of the signs of hypercholesterolemia. Well, in your blood vessels, you get something called the atheroma, and an atheroma is a plaque in that blood vessel wall. And typically, these plaques form as a result of oxidized LDL particles. We mentioned that before. So it's not just the cholesterol itself, it's the oxidized cholesterol that is then associated with inflammation. It's going to cause a lot of these problems, and it starts off with that atheroma. And then there's something called a xanthoma. These are plaques or nodules of histiocytes that are filled with lipid. Histiocytes are your dendritic cells of your immune system, and they get filled with those lipid particles in the skin, especially around the eyelids. And also an important thing here is xanthomas appearing on the eyelids are called xanthelasmas. And if you have a xanthoma on your eyelid, remember xanthelasma, it's a weird word. You can also have tendinous xanthomas, where you have lipid deposits in your tendon. Most common location is the Achilles tendon. And then the corneal arcus, is a lipid deposit in the cornea itself. It's a little bit nonspecific, but it still can definitely be a sign of hyperlipidemia. All right, so now let's move on to cholesterol synthesis. So we talked about absorption of fats, cholesterol through your gut, but your body can make its own cholesterol de novo as well. So what is the substrate for making cholesterol? Well, it's acetyl-CoA. So what is the rate-limiting enzyme of cholesterol synthesis? It's HMG-CoA reductase. Now don't confuse that with the rate-limiting enzyme for ketone synthesis. That's HMG-CoA synthase. And so what are the drugs that inhibit the HMG-CoA reductase? Well, it's the HMG-CoA reductase inhibitors, also referred to as statins, things like atorvastatin. So those are really the only three things that I want you to know for step one about cholesterol synthesis. It comes from acetyl-CoA, HMG-CoA reductase is the rate-limiting enzyme, and statins inhibit that enzyme. So now let's move on to some fatty acid metabolism. So fatty acids are just a carboxylic acid with a long aliphalic tail. Now most of the time they're found in triglycerides. Now a triglyceride is an ester with 3 fatty acids kind of attached to it. So fatty acids are small, and when they're not attached to triglycerides, they're called free fatty acids. Now let's talk about fatty acid synthesis. So the main player in fatty acid synthesis is the acetyl-CoA, which is again the precursor molecule to fatty acid synthesis. So where does a fatty acid synthesis actually take place? Well, it occurs in the cytoplasm of hepatocytes, and you're going to synthesize things like proteins and fats. Now, there are a lot of steps involved in fatty acid synthesis. I don't think it's super high yield to know every detail of it, but what I really want you to remember is the rate-limiting enzyme of this pathway, and that's going to be acetyl-CoA carboxylase. So acetyl-CoA carboxylase. So now let's move on to fatty acid degradation. So where is fatty acid degradation going to take place? It's actually going to occur in the mitochondria. So the beta oxidation, or the breakdown of acetyl-CoA into groups, occurs in that mitochondrial matrix. And from here you can get ketone buildup bodies, or you can also get move all this stuff into the TCA cycle. So you can either go to ketones or to TCA. Now the rate-limiting enzyme of fatty acid degradation is called carnitine acyltransferase 1. Sometimes referred to, just because we like things to be really complicated in medicine, it's referred to as carnitine palmitoyl transferase 1. So know both of those enzymes. Now what if you're deficient in that rate-limiting enzyme of fatty acid degradation? If you're deficient in that carnitine acyltransferase 1, sometimes also referred to as CAT1, well, you're not going to be able to get your long-chain fatty acids actually into the mitochondria. So you're going to get an accumulation of all these in your cytoplasm, and they can't be broken down because that machinery to break them down isn't even there. So that's going to cause things like weakness, hypotonia. You're also going to get a hypoketotic, a hypoglycemia. All right, that's going to be it for the video part of this. Go ahead and go through the end of session quiz, and let's go through those together now. First question, what deficiency causes familial hypercholesterolemia? So remember, that's going to be a deficiency in LDL receptors. Next, which apolipoprotein matches each of the following statements? This is the hard stuff. So first we have activates LCAT. Remember, that's going to be ApoA1. Next we have mediates chylomicron secretion. Remember, that's going to be ApoB48. Mediates VLDL secretion, that's going to be ApoB100. Cofactor for lipoprotein lipase, remember that's going to be ApoC2. And then mediates uptake of remnant particles, that's going to be ApoE. Next, what is the rate-limiting enzyme for each of the following metabolic pathways? Another tough one here. So fatty acid synthesis. Remember, the enzyme is going to be acetyl-CoA carboxylase. Next, we have beta oxidation of fatty acids. That's going to be carnitine acyltransferase 1. Ketone body synthesis, this is just so you don't forget this, is HMG-CoA synthase. And then for cholesterol synthesis, remember it's HMG-CoA reductase. Next question. Which group of medications inhibits the rate-limiting enzyme of cholesterol synthesis? So remember, it's going to be the statins. Those are going to be the HMG-CoA reductase inhibitors. All right, guys, that brings us to the end of the lecture. That's the end of Biochem 13. I hope you learned something. I'll see you next time.
Let's get warmed up for Biochem 14. First question, describe what light reflexes will be seen in both eyes if the right optic nerve is damaged prior to the pretectal nucleus. So this is also known as an afferent pupillary defect. So when you shine light into the right eye, there's no constriction of either the left or right pupil. But when you shine light into the left eye, both pupils constrict. Next, what are the major ECG changes seen with a myocardial infarction? So you can see ST segment elevation of at least 1 millimeter in 2 contiguous leads. You can see T-wave inversion. You can have a new left bundle branch block. You can have new Q waves, so Q waves that are at least 1 block wide or a third of the height of the total QRS complex. But remember, you only see these changes about 50% of the time when a patient's having an MI. So just because the ECG is normal doesn't mean you can rule out an MI. And the last one, what are the clinical features of neuroleptic malignant syndrome? So you're going to see delirium and mental status changes early on. You can see autonomic instability like tachycardia. Then you're going to see muscle rigidity, myoglobinuria, and hyperpyrexia. Okay, let's move on to the lecture.
Hello everyone and welcome to our Step 1 video on amino acids and nitrogen. You won't need a bunch of tryptophan to sleep through this one. Ha 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 ha. I kid. I mean, amino acids are fascinating. What with all the pathways and enzymes and cycle thingies. Woohoo! Okay, really, go get some coffee, an energy drink, or maybe that totally legit methamphetamine that your doctor gave you for your intermittent attention problem. Yeah, I know. All right, so now let's talk about protein and amino acid metabolism. Now, as always, biochem is a bit rough. You may need to spend a little extra time after the video to memorize a lot of the enzymes that we're going to talk about today. So first we have our amino acid. Let's talk about these. What are the essential amino acids? What does it mean to be essential? Are they more important? Well, the answer is not really. It's a little bit of a misnomer. But the essential amino acids are the amino acids that need to be acquired through the diet. We can't synthesize these at all. We have to eat them. Now, a great mnemonic for essential amino acids is Private Tim Hall. So the Private Tim Hall mnemonic is PVT, and then the TIM for Tim, and then Hall is all the letters except for the A. So PVT for private is going to be the phenylalanine. The V in private is going to be the valine, and then T is for threonine. And then we move on to TIM, TIM. That goes for tryptophan, isoleucine, and methionine. And then Hall is for histidine, leucine, and lysine. So a good way to remember all that, use that Private Tim Hall mnemonic. So this now brings us to the concept of acidic versus basic amino acids. So let's talk about your basic ones first. So lysine and arginine are particularly basic. They have an extra ammonia group, and histidine is also a basic amino acid as well. Arginine and histidine are particularly important during periods of growth. And then arginine and lysine are important for nuclear localization signals. Arginine and lysine, again, are found in high concentrations in histones. Why is that important? Well, because of their positive basic charge, they can interact with the negatively charged DNA. And then your acidic amino acids are aspartate and glutamate. And that's actually a little bit easier for me to remember because they're also referred to as aspartic acid and glutamic acid. So they're negatively charged at the body pH, whereas the basic amino acids tend to be positively charged. Now, one exception to this rule is that histidine, although it's considered a basic amino acid, has no charge at body pH. All right, so now let's get to some harder stuff. Derivatives of amino acids. So let's talk about those derivatives. What are some of the interesting compounds or things that your body can make with amino acids? Now again, this is a pretty high, probably a 5-star topic. You're gonna have a question about amino acid derivatives. So first let's start with phenylalanine. Phenylalanine plays an important role in the catecholamine synthesis pathway. The amino acid phenylalanine is essential for the generation of tyrosine. Tyrosine is found in high concentrations in thyroglobulin, and then you iodinate those tyrosine residues to make thyroxine, so your thyroid hormone, so very, very important. Now, tyrosine can also be converted to DOPA, and DOPA can be converted to melanin. Now, where in your brain might you have high levels of melanin because of high concentrations of DOPA and dopamine? That would be your substantia nigra, which is damaged in patients with Parkinson's disease. They lose the dopaminergic cells, so they don't make dopamine and they don't make melanin in the substantia nigra. Now, another disease related to this is albinism. If patients can't convert DOPA to melanin, then that results in albinism. So let's move further down here. DOPA is converted to dopamine. Dopamine to norepinephrine is by something called dopamine beta hydroxylase, and that's with the aid of vitamin C. This enzyme is also called dopamine beta monooxygenase, so don't get confused by that. You'll probably want to add that enzyme to your progression in your study guide there. Now, norepinephrine to epinephrine is going to be done with the help of cortisol and something called SAM. We'll talk about that here in just a second. So let's talk about a little bit more about these enzymes and how they're important. Well, remember, phenylalanine to tyrosine is accomplished by phenylalanine hydroxylase. And if you're deficient in that enzyme, you get PKU. And we'll talk about that in the next video. A lot of these will get a little bit more play in the next video when we talk about the disease states. Not only is phenylalanine hydroxylase needed, but the tetrahydrobiopterin is also required when converting phenylalanine to tyrosine. If either are deficient, then you're going to get PKU. Now, tyrosine goes to DOPA by tyrosine hydroxylase, and then the third enzyme of this pathway that I want you to know is DOPA decarboxylase, and that converts DOPA to dopamine. And that particular enzyme, DOPA decarboxylase, is inhibited by carbidopa, a medication we use to treat Parkinson's disease. Now, dopamine is going to be converted to norepinephrine by dopamine beta hydroxylase with that aid of vitamin C we talked about before. Now, there's something called dopamine beta hydroxylase deficiency where the patient has excess dopamine but a deficiency of norepinephrine and epinephrine. Patients will present with nasal congestion, ptosis, and orthostatic hypotension. And then norepinephrine is converted to epinephrine, and cortisol is going to facilitate that conversion, as not the actual enzyme, but it helps move it along, of that norepinephrine to epinephrine. So it actually kind of makes sense that the stress hormone cortisol would facilitate that generation of a flight or fight hormone, epinephrine. Now, another cofactor here that you should probably know is SAM. We talked about that before, and that stands for S-adenosyl L-methionine. So SAM is another important cofactor there. Now, what happens when you break these things down? Well, dopamine gets broken down by the enzymes monoamine oxidase and catechol O-methyltransferase, and that's going to lead it down a pathway to HVA. Norepinephrine gets broken down to something called VMA, and then epinephrine to something called metanephrine. Important stuff there. All right, so that all started with phenylalanine. What can you make out of the other amino acids? Well, let's start with arginine. Arginine is used to make 3 different important compounds. It can be used to make creatine, which is a high-energy phosphate carrier that replenishes ATP. It can transfer its phosphate group to ATP. Now, when ATP starts to run out, you have this kind of backup energy metabolite of creatine phosphate that's going to rapidly phosphorylate ADP back into ATP. So it's very, very important. Arginine is also important for the generation of urea, which we'll talk about more in just a minute with the urea cycle. And then arginine can also be converted into nitric oxide as well. And nitric oxide is a smooth muscle relaxer, and it's essential in vasodilation. All right, so now let's talk about several amino acid derivatives that are dependent upon vitamin B6 as a cofactor. So many of our reactions use vitamin B6, and you'll notice in a second that without B6, we would have some potential serious problems. So first of all, B6. Tryptophan is converted to niacin with the help of B6, which is particularly important in the body for the generation of NAD and NADP. So these are essential in oxidation and reduction reactions. Now, tryptophan is also essential for the generation of serotonin, which is a precursor to melatonin. And just as an aside, which gland makes melatonin? Remember, it's the pineal gland. Now, histidine is converted to histamine. Again, you need vitamin B6 for that as well. And then B6 is also needed for the conversion of glycine to porphyrin. And porphyrin is converted to heme. Now, the rate-limiting enzyme for heme synthesis is aminolevulinate synthase. And then the amino acid glutamate is essential for the production of GABA, again with the help of B6 again. And GABA is the main inhibitory neurotransmitter of the central nervous system. So with B6 deficiency, you're not going to have as much GABA around, and that can actually result in a lack of inhibition, which may actually lead to seizures. And then also glutamate is important for the generation of glutathione. Glutathione is an important antioxidant. So B6, super important. All right, so we use our amino acids to make all those substances that we just mentioned. And of course, we put amino acids together to make enzymes and receptors and proteins and lots of other complex molecules. But whenever you break down amino acids, there has to be a mechanism to get rid of the nitrogen. So yes, it's time to talk about the urea cycle. So urea is made in the liver. Now, first of all, you need to know what urea looks like. It's a carbon molecule with a double bonded oxygen and then the NH2 on either side of that carbon. So the rate-limiting enzyme for the urea cycle is carbamoyl phosphate synthetase 1. And it's found in the mitochondria. And that might actually sound a bit familiar because carbamoyl phosphate synthetase 2 was the rate limiter of pyrimidine synthesis. So again, just to contrast, with CPS1, it's used in the urea cycle, it's found in the mitochondria, and it gets its nitrogen from ammonium. CPS2 is used in pyrimidine synthesis, and it's found in the cytosol, and it gets its nitrogen from glutamine. So be aware, they're actually pretty different. Now, I don't think you need to get bogged down in the different enzymes and substrates of everything in the urea cycle. I would know CPS1 in the mitochondria as the rate limiter, and I would also know one other enzyme, ornithine transcarbamylase is another very important enzyme in the urea cycle. So let's go actually a little bit deeper into that one. What happens if you're deficient in ornithine transcarbamylase? That is the most common urea cycle disorder. So this is an X-linked recessive disorder. And if you don't have that enzyme, then you can't undergo the urea cycle. You're going to build up nitrogen and ammonia in the body. Now, this is problematic, obviously, and this becomes evident in even just the first few days of life. All this excess carbamoyl phosphate is converted to orotic acid. So you find orotic acid in the blood and in the urine. Your blood urea nitrogen is going to be actually decreased because the urea cycle doesn't work. You can't make urea. So BOM that we'll see on a lot of comprehensive metabolic panels in the rest of your life is actually an indicator that the urea cycle is working. If it's elevated, you're probably making that urea, but you're not excreting it via the kidneys very well. If BON is decreased, it's probably that you're not making urea. So with ornithine transcarbamylase deficiency, you're going to have symptoms of hyperammonemia, so elevated ammonium levels. You can have elevated ammonia levels either from a particular urea cycle enzyme deficiency or because the cell responsible for the urea cycle is damaged, and this is specifically the hepatocyte. So if you have liver disease, that can cause that hyperammonemia as well. So what are the symptoms of hyperammonemia, that elevated ammonia? Patients will have slurring of speech, they'll have somnolence, vomiting, cerebral edema, and even a blurring of the vision as well. Now, we refer to this constellation of symptoms as hepatoencephalopathy. So again, you can see this with liver disease or if you have a specific urea enzyme deficiency. And again, the most common urea enzyme deficiency is the ornithine transcarbamylase deficiency. So how do we treat this? Well, since this is a nitrogen processing problem, then we try not to increase the nitrogen load by eating a low protein diet. Now, if it's a urea cycle problem, you can use phenylbutyrate, which binds certain amino acids like glycine and glutamine and helps to excrete these amino acids. Benzoate is also a list on the list of drugs that can be used as well. Biotin can also be helpful because it actually stimulates ornithine transcarbamylase as well. All right, so that gives you the first taste of amino acids. We'll go over a little bit more in our next lecture on some of the more specific pathologies. But now it's time for that end of session quiz. I want you to try to answer those on your own, and then we'll go through them together. All right, first question. What are the essential amino acids? Remember, we use that mnemonic private Tim Hall. So PVT is for private. So that's phenylalanine, valine, and tryptophan. And then Tim is for the T. First T is threonine, isoleucine, methionine. And then we have the Hall, histidine, leucine, and lysine. So those are the essential amino acids. We have to eat those because we can't make them. Next, what amino acid is a precursor for the following molecules? This is the tough stuff. So first we have histamine. Remember, the precursor is histidine. Next we have porphyrin and heme. So remember, the precursor there is glycine. Nitric oxide, the precursor there is going to be arginine. The neurotransmitter GABA, so the precursor there is glutamate. S-adenosyl methionine, the precursor there is just plain methionine. And the precursor for creatine is arginine. Next, compare CPS1 to CPS2. So that's the carbamoyl phosphate 1 and 2. So CPS1 is part of the urea cycle that we just talked about. And that part's going to take place in the mitochondria, and it gets its nitrogen from ammonium. CPS2 is involved in pyrimidine synthesis. It's found in the cytosol, and it's going to get its nitrogen from glutamine. So very different there. All right, guys, that brings us to the end of the video. I hope you learned something. I'll see you next time.
Let's go over the warm-up. First question, what dermatologic finding is seen in each of the stages of syphilis? So in primary syphilis, the skin finding is a painless chancre. In secondary syphilis, you can see a maculopapular rash on the palms and soles, and also condyloma lata. And then with tertiary syphilis, you can see gummas. Next, which antihypertensives are commonly used during pregnancy? So the mnemonic is hypertensive moms love nifedipine, which stands for hydralazine, methyldopa, labetalol, and nifedipine. Then the last question, what does each of the following cells of the GI tract secrete? So the G cells of the stomach secrete gastrin, the I cells secrete CCK, the S cells secrete secretin, the D cells secrete somatostatin, and then the parietal cells of the stomach secrete both gastric acid and intrinsic factor. Now let's move on to the lecture.
Hello everyone, and welcome to our Step 1 video on amino acid disorders. It's time to find out which of you have a potentially devastating disease and which of you just naturally smell musty and have funny looking pee. Okay, so amino acid disorders. Today we're going to first cover PKU, phenylketonuria. Now PKU is definitely high yield for Step 1. It's a 4-star topic. You're very likely to have a question on PKU. So what is it? Well, there are two ways to have PKU. You can either be deficient in the enzyme phenylalanine hydroxylase, which converts phenylalanine to tyrosine, or you can be deficient in the cofactor for phenylalanine hydroxylase, which is a hard one, tetrahydrobiopterin. And so in this disease, just like with any other metabolic disorder, the next product down the line is going to be deficient, and in this case it's going to be tyrosine. So tyrosine actually becomes an essential amino acid at this point for these patients because you can't make it on your own anymore. You have to bring it in from the outside. And also, you don't want to give these patients things like phenylalanine because it's just going to build up. That's going to be the other big problem. They can't process phenylalanine, and it's going to result in something called phenylketones. So what are phenylketones? Well, phenylketones include things like phenylacetate, phenylactate, and phenylpyruvate. So with PKU, these phenylketones build up when you eat foods rich in phenylalanine. But realize that the actual neurotoxic effects are actually related to the high levels of phenylalanine and not actual the phenylketones. And as a result, this can cause things like growth retardation, intellectual disability, seizures, fair skin, eczema, and then the all-important musty body odor. And that's a big giveaway on a lot of your test questions. Now, PKU is a disease that you want to diagnose as soon as possible so you don't get these manifestations. So for this reason, we actually screen infants at 2 to 3 days of life, very early on. Now, why can't you screen these infants just right at birth or prior to birth? Well, you have to actually wait for these phenylketones to build up to a certain measurable level in the urine. So after delivery, moms can actually provide tyrosine in utero to the baby. But after delivery, as the baby is getting phenylalanine in the breast milk, it's going to start building its own phenylketones in that urine. And then you can actually start testing for these in the urine as well. So how do we treat this? Well, patients should avoid phenylalanine to start with. So what is high in phenylalanine? Well, a lot of great things, unfortunately. Aspartame, which maybe isn't that great, it's just an artificial sweetener. But the other things are really important. Things like dairy products, meat, fish, chicken, eggs, beans, nuts. So a lot of great things have phenylalanine in them. And then again, you need to add tyrosine. That was our deficient problem in this diet because they will become tyrosine deficient. Again, it also may not be an enzyme deficient problem. It could be that cofactor. Remember, it's that tetrahydrobiopterin that could be deficient in this situation. So that's going to be essential as well. So supplementation of that tetrahydrobiopterin could be necessary as well. And this needs to be accomplished within really the first 3 weeks of life in order to prevent a lot of the characteristic effects of this disorder. Now, what would happen to the infant of a pregnant mom that has PKU, that's been managing her PKU her whole life, and then for whatever reason, while she's pregnant, decides that it's too difficult and she doesn't want to manage it very well? What would happen in that case? Well, the infant would have microcephaly, intellectual disability, growth retardation, congenital heart defects as well. So these phenylketones and phenylalanine are going to be very toxic to that developing fetus. All right, so let's move on to another amino acid disorder called alkaptonuria. And alkaptonuria causes something called ochronosis. And so what is the enzyme that's deficient in this situation? The enzyme here is homogentisate oxidase, or also known as homogentisic acid oxidase. So these patients can't degrade tyrosine normally. Now this is an autosomal recessive disorder, but it's not quite as severe as PKU. They have this ochronosis taking place where they have homogentisic acid, and that's going to build up in the urine and in the connective tissue. And homogentisic acid is a brown pigment, so the connective tissue becomes dark. The sclera can become darkly pigmented with spots as well, and the urine's going to turn black as it's exposed to air. And they can have arthralgias again because of that homogentisic acid. It's being deposited in the connective tissue in the joints, and that's actually toxic to the cartilage and can cause pain. Now, one way to remember alkaptonuria, remember alkaptone has dark connective tissues, dark sclera, black urine thickening. So I think of Al Capone, alkaptone, kind of wearing a black dark suit, and that's one way to remember that. A little bit of a stretch, but I think it's so good. Now, there isn't really much in the way of effective treatment. Vitamin C supplementation might be helpful. Dietary restriction of phenylalanine and tyrosine can be tried. But again, treatment is not very effective at this point. So now let's move on to albinism. So albinism is a congenital disorder where patients have complete or partial absence of pigment in the skin. So patients have the normal number of melanocytes, but there's a problem with melanin production. So remember that vitiligo is a skin depigmentation, but that's due to autoimmune destruction of melanocytes. So they have fewer melanocytes. But again, albinism, the melanocyte number is normal, but they can't produce enough melanin. So why is that? So here you can either have a tyrosinase deficiency or a defective tyrosine transport. If you can't get tyrosine into the cell, then it can't be metabolized into melanin. And so those are two ways that you can have albinism through the amino acid pathway. All right, so another amino acid disorder is homocystinuria. So like the name implies, this is having homocysteine in your urine. So why would you have this? Well, homocysteine can be converted to a cystathionine, and then cystathionine gets converted to cysteine. Now, homocysteine can also be converted to methionine. Now, you can have homocystinuria if you have problems with either of these processes. Now, the conversion of homocysteine to cystathionine requires the enzyme cystathionine synthase, and that requires cofactor B6. So, homocystinuria can be caused by a deficiency of cystathionine synthase or by a decreased affinity of that cystathionine synthase for that peroxyl phosphate, that B6 cofactor. So, if the enzyme doesn't interact effectively with that B6, then it's not going to work either. Now, that other pathway and problem can occur if you can't convert homocysteine to methionine. So if you have a deficiency of homocysteine methyltransferase, you can also get homocystinuria as well. So as you accumulate more and more homocysteine, this is going to build up in the urine, and that's going to be where you're going to get your diagnosis most of the time. So what are the clinical manifestations of this? Well, in addition to homocysteine in your urine, these patients will have intellectual disability, tall stature, osteoporosis, kyphosis, atherosclerosis, and subluxation of the lens of the eye. Now, in what other disorder might you have that subluxation of the lens? Well, think of Marfan syndrome. So two genetic diseases or disorders in where you're going to have that lens subluxation. Now, homocystinuria and Marfan syndrome are still a little bit different. The one difference between the two is that we see a downward lens dislocation in homocystinuria, and then in Marfan syndrome, we see an upward lens dislocation. So how do we treat homocystinuria? Again, you have to know the metabolic pathway. You have to get around that metabolic pathway. If it's a cystathionine synthase problem, then you're going to decrease methathione in the diet. You're going to increase cysteine in the diet because you might get deficient at that point. You're going to increase B12, folate, and especially B6. Remember, B6 is that cofactor, very important. So diet should be high in B12, folate, B6, and cysteine to bypass that pathway, and then low in methionine. Now, we're talking about homocysteine. I also want to mention another substance called SAM, or that S-adenosylmethionine. Now, why is S-adenosylmethionine important? And this is more of a biochem question rather than a problem with a disorder. Well, SAM transfers methyl units, and the transfer of methyl units is particularly important for the conversion of norepinephrine to epinephrine, and also for the synthesis of phosphocreatinine. And so for those two interactions, you need SAM. Now, what else do you need to know about the S-adenosylmethionine? Well, it's made from ATP and methionine. So that's how you get SAM. And the regeneration of methionine is dependent upon things like B12 and folate. So if you don't have B12 and folate around, you're not going to have very much methionine. And without methionine, you can't make that S-adenosylmethionine. So if you can't have S-adenosylmethionine, you're not going to be able to make things, as we said before, those phosphocreatinine, your epinephrine from norepinephrine. So that's what you really need to know about SAM. There's this long process of you're deficient in one thing, it's going to mean you're going to be deficient in another thing. Now, the next amino acid disorder I want you to know about is cystinuria. Now, cystinuria is a defect of the renal tubular amino acid transporter for what we call COLA, C-O-L-A. And this is a mnemonic for cysteine, ornithine, lysine, and arginine. Now, this transporter is normally found in the proximal convoluted tubule of the kidney. But with cystinuria, you have a defect of that COLA transporter in the proximal convoluted tubule. So why is cysteine in your urine a problem? Well, you can form cysteine kidney stones. And the way you want to prevent this and to treat this is with something called acetazolamide. So acetazolamide is a carbonic anhydrase inhibitor that is going to alkalize the urine and prevent the development of those kidney stones. Now, one thing to clear up is the difference between cysteine and cysteine, and they're spelled differently. So there's the C-Y-S-T-I-N-E, and then there's the C-Y-S-T-E-I-N-E. So one with an E, one without an E. So cysteine without an E is an amino acid, and it's formed by two cysteines with an E bound together by a disulfide bond. So it's probably not too important, but don't get confused. They're not actually the exact same thing. One is made up by two of the other. So sometimes I think we're our own worst enemy when we're naming things in medicine. Now, another amino acid disorder that puts weird stuff in your urine is called maple syrup urine disease. Now, in this disorder, the urine is actually going to smell like maple syrup. And the reason why is because it's filled with the branched chain amino acids isoleucine, leucine, and valine. Now, one mnemonic that you can use to help you is I love maple syrup. So the I, that L, that V to help you remember the maple syrup. So the I is for isoleucine, L is for leucine, and V is for the valine. Now, this comes about because of a deficiency in the enzyme complex called the branched chain alpha keto acid dehydrogenase complex. That's a long one. Now, it's supposed to be breaking down all these branched chain amino acids. And if the enzyme is deficient, these branched chain amino acids don't get degraded like they're supposed to, and they build up in the urine and in the blood, along with the corresponding branched chain alpha keto acids. Now, this causes a lot of the same symptoms we've seen in all these things: intellectual disability, CNS defects, high mortality rate as well. Now, what about Hartnup disease? So this is the last disorder that we're going to cover. Hartnup disease is an autosomal recessive defect of a transporter in the intestine and the kidneys, and that's supposed to be transporting neutral amino acids like tryptophan. And this causes tryptophan to be excreted in the urine and not be absorbed in the gut. So what happens when you don't have tryptophan? Well, if you don't have tryptophan, your body can't make niacin. And when your body can't make niacin, you get pellagra. Now, we'll talk about pellagra in more detail with the vitamin deficiencies, but this is a vitamin B3 deficiency. And when you're deficient in vitamin B3 or niacin, it causes that triad, the big Ds. So the three Ds: dermatitis, diarrhea, and dementia. So these are called the big Ds, or the three Ds of vitamin B3 deficiency. Remember, it's going to be dermatitis, diarrhea, and dementia. Okay, so whenever we have unnecessarily complex names to remember, that means that we need to review all this in Lewis notes. Hello everyone, welcome to another Lewis Notes. We're talking about amino acid diseases here. We've got our big amino acid diseases here, and what we're looking at are the red here, we have the enzymes that are deficient, and then in the blue we have some high yield stuff that I want you to remember as well. So write this stuff out with me. It'll be an easier way of remembering all this stuff. So first we're going to start with our red enzymes here. We're going to go backwards. First we have the homogentic acid oxidase. So you look at that and you think, whoa, they both have homo in them, they must go together. This actually does not go with homocystinuria. This actually goes with alkaptonuria. So remember the homogentic acid, all that stuff goes with alkaptonuria. Next we have the alpha keto acid dehydrogenase. And that one is going to be going with our maple syrup urine disease way down here. So lots of long words on that one. Tyrosinase is going to be going over here with albinism, so you can't make your melanin at that point. Decreased tryptophan. So if you have decreased tryptophan, you do have to worry about heart disease. That's what is going to lead to that one right there. Phenylalanine hydroxylase. That one's pretty easy because the phenylalanine makes you think of PKU, phenylketonuria. Cystathionine synthase, that's going to be over here by homocystinuria. You could get confused with that one and think it goes with cystinuria, but it actually doesn't. Next we have B6, and that B6 is going to go along with homocystinuria again. Remember that was another kind of cofactor that went with that one. The big cola, that mnemonic that was used to help remember where this is happening in the kidney, that's going to go with cystinuria. And then that tetrahydrobiopterin, that really super long one, is another cofactor that then goes along with PKU. All right, so now let's take a minute and let's look at the high yield stuff that you got to remember in regards to all this stuff, because you can remember the enzymes, but you have to also remember sort of where this is taking you and all this type of stuff. So the first one we have niacin. So niacin is going to be associated down here with that heart disease. Remember that will decrease tryptophan, you're not going to be able to make much niacin, and then you get your 3 Ds. Remember what the 3 Ds were? Dermatitis, diarrhea, dementia. Sweet smelling urine, that one's pretty easy because it's just in the name there. So maple syrup urine disease, that one's pretty easy. The musty smell, always go with PKU if you see musty smell on a test right there. Dark urine, if you see dark urine, you're always going to want to go to alkaptonuria, especially if that urine's been sitting out. Also dark sclera, also dark connective tissue and that sort of thing. Increased skin cancer, very easy to go here with albinism. Lens dislocation, this one's a hard one to remember in my opinion. And remember that's going to be that downward lens dislocation. So that's going to be with homocystinuria. And then the cystine stones, that's going to go along with that cystinuria. Remember you're going to be getting a lot of those types of stones in regard to that, and you can use acetazolamide to help treat that sort of thing. All right, so that's the high yield stuff. Those are the high yield enzymes. I hope you can remember all that type of stuff, and then you'll be okay. All right, that's going to be it for the amino acid disorders. Let's go ahead and go through the end of session quiz together. First question here, a full-term neonate feeds poorly, is hyperactive, and has a musty odor. What is the diagnosis? So the musty odor is something you got to remember here. That's going to be your clue for PKU, phenylketonuria. Next question. A patient with PKU should have a diet low in phenylalanine. What other dietary modifications should a patient with PKU make? So first, you want to probably increase tyrosine because you're going to be deficient in that because you're not producing very much of it. And then you're also going to want to replace the tetrahydrobiopterin cofactor if that is also deficient. Next question. A middle-aged man has dark spots on his sclera and has noted that his urine turns black when left sitting for a period of time. I don't know why he's doing that, but it's happening. What is the diagnosis? So this is the easy one. That's going to be that alkaptonuria. Next question. What is the underlying cause of maple syrup urine disease? So this is going to be a deficiency of the branched chain alpha keto acid dehydrogenase complex. That's the long one to get. So you have alpha keto acids in the urine because you can't break down those amino acids like leucine, isoleucine, and valine. Remember, these are the branched chain amino acids. All right, guys, that brings us to the end of our lecture, and I hope you learned something. I'll see you next time.
Hello there. Let's play a little game. I've got a treat for you. I'll give you 3 clues. See if you can guess the condition. 1. It is caused by deficiency of the enzyme alpha keto acid dehydrogenase. 2. It leads to a buildup of the 3 branched amino acids isoleucine, leucine, and valine. 3. Last hint. It can cause mental retardation and death. That's right. It's maple syrup urine disease. The name comes from a sweet, distinct maple syrup-like smell from the patient's urine. Oh, honey, you want some more? More? Oh, where are you going? Oh, well, more for me.
Let's go over the warm-up review. First question, what's the pathogenesis of glaucoma? Glaucoma is caused by increased intraocular pressure, which damages the optic nerve. In open-angle glaucoma, there's impaired outflow of aqueous humor because the canals of Schlemm and the trabecular meshwork become plugged up. In closed-angle glaucoma, the lens of the eye moves anteriorly up against the iris and blocking the pupil so that the aqueous humor can't flow from the posterior chamber of the eye to the anterior chamber. And in either case, the aqueous humor kind of builds up and the intraocular pressure increases. Next, which infectious agent is the cause of pneumonia based on each of the following lab tests? So gram-negative rods in a neonate with pneumonia, think about E. coli. Gram-positive cocci in a neonate with pneumonia, think about group B strep. Gram-positive cocci in pairs is classic for strep pneumoniae, and gram-positive cocci in clusters is classic for Staph aureus. And the last question, what's the classic presentation of aortic dissection? It's going to be tearing chest pain that radiates through to the back. Now, what chest x-ray findings would support the diagnosis? Mediastinal widening on the chest x-ray. And what antihypertensive class is the preferred treatment? It's going to be beta blockers. All right, now let's move on to the lecture.
Hello everyone and welcome to our Step 1 video on minerals. Have I told you lately how much I hate modern day vampires? First, who thought it was a good idea to make vampires mopey emo teenagers? All you hear is whine, whine, whine. No one understands how hard it is to be a vampire and girls don't like me. Get over it, Chompy. Remember the good old days when vampires were scary? They slept in coffins, and your best defense was throwing a clump of garlic their way. And when they went out into the sun, they died. They didn't sparkle. On what planet do vampires sparkle in sunlight? If a vampire goes out into the sun, they need to catch on fire and explode, or I'll accept crumbling into a pile of dust. So, unless your vampire is a pasty 48-year-old scary man with an Eastern European accent and a cape, then I want nothing to do with him. There's one exception for puppet vampires that teach me to count to 10. That guy's okay. All right, let's get going. So today we're talking about minerals, and the first one we're going to hit today is iron. So where do we find iron in the body? Well, when you think about iron in the body, you typically think about RBCs, and then that iron that's going to be found in hemoglobin. You also have iron in myoglobin, so in your muscle cells. So where do we store excess iron though? Well, you're going to store it in the liver, you're going to store it in the spleen, and then in the bone marrow. Those are going to be the biggest places that you're going to find iron stored. Now, if you don't have enough iron stored up, that will lead to microcytic anemia, and we're going to cover that a lot more in the hematology sections. Now, if you have too much iron, then that can be a problem as well. So that's going to lead us to our next section, iron poisoning. So iron poisoning used to be one of the leading causes of fatality from toxicologic agents in children about maybe 20 years ago, maybe further. Now the most common scenario was that children would get into mom's purse or the medicine cabinet and take prenatal vitamins or iron supplements that maybe someone in the family had received for anemia. But now we have much better packaging of iron-containing pills, so that doesn't happen much anymore. But what would happen if you were to have iron poisoning? Well, iron causes a lot of peroxidation of lipid membranes and also generates a lot of free radicals. So iron can be very, very damaging to cells for that very reason. Now, what are the symptoms though? Well, initially you might have GI problems, specifically gastric bleeding, because the first place that iron pills are going to settle in is in the stomach, and it's going to cause a lot of damage to those cells, which then will lead to that bleeding. And you can either vomit blood, so you could have hematemesis, along with having maybe a lot of abdominal pain, or you may not throw up and it may progress through your GI tract, and in either case you can have hypovolemic shock and even die if that bleeding is severe. Now let's look at some timelines. About 6 to 72 hours after ingestion, you can have a metabolic acidosis. Now you may be talking or thinking, why wouldn't you have metabolic alkalosis if you're vomiting? Well, in the very acute setting, if you're throwing a whop, then you probably would expect to find that. However, because now we're talking about maybe a little bit longer term or chronic toxicity, and when we say chronic we mean hours to days, you are going to have more of a metabolic acidosis. So be sure to remember this important finding. And then about maybe 2 to 8 weeks after the ingestion, you can have scarring of the GI tract. So areas that were damaged by those free radicals generated by iron can be scarred. And that can lead to obstruction of the GI tract. And again, that would take place 2 to 8 weeks after ingestion. So now let's talk a little bit more about iron. Let's talk about iron proteins. So this is involved in the storing and the transporting of iron in the body. So this is in your study guide. The first thing is ferritin. What is ferritin? Well, ferritin is an iron protein complex of ferric acid and apoferritin, and it's the cellular storage form of iron. So iron is stored inside of your cells as ferritin. So ferritin is actually in very high concentrations in your hepatocytes. So a lot of that iron, as we said before, is in the liver. It's bound to ferritin. And ferritin is also what we call an acute phase reactant. So what does that mean? Well, when there's a lot of inflammation, the hepatocyte is going to put more ferritin into the circulation. Well, why would it do that? Well, iron is actually a substance that bacteria really want to find. They can use it. So by binding up all that iron in your body, it's actually able to remove iron from the circulation so bacteria can't obtain it. So it's actually kind of a way of fighting off infections on a small level. So this is why your hepatocytes would then put that ferritin in there to help fight all that off. But again, your ferritin level in your serum, if it's increased, that could be a sign of inflammation or infection. Now let's talk about transferrin. So transferrin is a protein that binds ferric molecules and transports them through the plasma. And it's also synthesized by the liver. So it's a transport protein. Now if you want to get iron from the liver to another part of the body, say the bone marrow, then you would do that with transferrin. Trans kind of makes it easy to remember. Now the half-life of this molecule is about 8 days, and it's increased in cases of iron deficiency because your cells are hungry for iron. So they're looking for this transport molecule to deliver more iron as soon as it's available. But in the case of iron deficiency, there may be more transferrin, but it's not bound to any iron. So the transferrin binding capacity would actually be very high. It has a high capacity for binding in states of iron deficiency. All right, so that's enough about iron. Let's move on to zinc. So zinc is essential for at least 100 different enzymatic activities inside the body. Now, a couple that I want you to remember and for me to mention are the zinc fingers that are transcription factors for DNA transcription. If you don't have zinc, you're not going to be able to transcribe your DNA template to make proteins very well. So in states where you're trying to make a lot of proteins, say in a healing state where you're trying to make new skin, you would need zinc in your body to be able to transcribe new proteins, to be able to generate more collagen in that area for the healing process. So zinc is something that we commonly supplement in patients that are healing or patients that have a hard time healing. Zinc is also helpful for the immune system. Zinc is essential for carbonic anhydrase. So maybe you've seen that enzyme taking CO2 and converting it to a bicarb. Now we see that in the renal tubules and you see that in the red blood cells as well. Carbonic anhydrase is everywhere in the body essentially, and it requires zinc as well. Now the enzyme lactate dehydrogenase is also an enzyme that requires zinc as well. Now what happens if you don't have enough zinc? Well, you can have lots of problems. We just mentioned multiple things require zinc. You can have delayed wound healing. You can have decreased body hair. You can have decreased facial hair. You can have hypogonadism. You can have anosmia, which is decreased smell. Dysgeusia, which is decreased taste. You can have decreased immune responses. You could have a rash, and this rash can be around the eyes, around the mouth, around the nose, and the anus. This is called acrodermatitis enteropathica. Zinc deficiency will also cause anorexia and diarrhea. It can cause growth retardation in children, depression and depressed mental functioning, impaired night vision even. So in a similar way that vitamin A deficiency can impair your vision, zinc deficiency can also impair your vision. So there's actually specific enzyme supplements for eye health, and in those supplements there's a lot of vitamins A, E, and C, and also a lot of zinc. And then zinc deficiency can also cause infertility, so you need zinc in order to be fertile. Next, let's move on to calcium. So we're going to just briefly touch on calcium. We talk about in detail in our endocrine section, so I'm just going to kind of hit the highlights of a few things in calcium here. So what's important with calcium? PTH. So PTH, or parathyroid hormone, it's the most important regulator of calcium metabolism. Vitamin D is also very crucial in calcium metabolism. If you have excess PTH, then you're going to have excess calcium or hypercalcemia, and that's going to cause a whole constellation of symptoms that you might remember by the mnemonic stones, bones, groans, and psychiatric overtones. Now when your calcium is very low, that's called hypocalcemia, and one clinical sign is that Trousseau sign, where you tighten the blood pressure cuff and you get a carpal muscle spasm. So your muscles become more spastic and hyperreflexive. You can also have a Vostick's sign, where you tap the cheek and you have a facial muscle spasm. And a lot of calcium is going to be stored in the bone, not only for structural support, but also because it's essential that if you need a ready supply of calcium as a reservoir, then you can actually get that out of the bone. So you really can't do without calcium. Very important for muscle contractions, it's important for neurotransmitter release, it's important for platelets to function, for clotting cascades to be activated, it's important for intracellular processes, acting as an intracellular mediator. Without calcium, things like glucose and ATP are essentially worthless. So calcium, big deal. And again, we cover that in endo a lot more than we will here. Moving on to lead, and we're going to hit lead poisoning as well, and we're going to talk about some toxic metals, and starting with lead here. So what are some of your earliest signs of lead poisoning? Well, you're going to get things like decreased IQ, you can have hearing problems, growth impairment, you can have impaired peripheral nerve function. So one of the earlier signs of lead poisoning is peripheral nerve neuropathy, and that can lead to things like wrist drop or foot drop. So if you see wrist drop and foot drop, think about lead poisoning. What else? Lead also interferes with normal remodeling of cartilage. So if you have an area where you're remodeling cartilage, for instance, growing bones, then you're going to get lead lines in those bones. Now on the test, they may give you a radiographic image showing bones of a patient along with the clinical presentation of lead poisoning. So be sure to be able to put all those clues together. You can also get lead lines in the teeth, and you can also get them in the gum junction as well. And those are also called Burton's lines. Now, severe lead toxicity can also cause decreased hemoglobin synthesis that can then lead to anemia. So patients may have colic or abdominal pain with this as well. And the mechanism here is that lead inhibits ferrochelatase and ALA dehydratase, which leads to decreased hemoglobin synthesis. And then lead can also cause nephropathy, encephalopathy, and then even death. You should also know that on a peripheral blood smear, lead toxicity can result in basophilic stippling of red blood cells. Lead inhibits rRNA degradation, which causes RBCs to retain segments of rRNA, and this results in that basophilic stippling appearance. Lead can also cause ringed sideroblasts in the bone marrow. A ringed sideroblast is a red cell precursor containing mitochondria that are filled with iron. So how do we treat lead toxicity? Well, there are several different regimens published in various books. First of all, for mild lead toxicity, you're probably not going to need to treat anything at all beyond removing the source of lead exposure. But if the lead levels are moderate or high, that they're causing symptoms, then you would treat it with a chelating agent. Either probably you're going to use Succimer or EDTA, probably the calcium version of EDTA. And that's the same for both adult and children. And then if you have pediatric patients with very severe lead toxicity, then you would potentially use dimercaprol, maybe dimercaprol plus Succimer. So that's what you need to know about lead. Let's move on to mercury. Mercury poisoning, not as often seen now, and we're going to cover this in the study guide. Mercury accumulates in the kidneys and in the brain. Got to remember that, kidneys and brain. So it causes peripheral neuropathy, neurological problems like a tremor, neuropsychiatric problems like excitability and insomnia. And it can also cause something called acrodynia, which is the peeling of fingertips with the skin. So that's something to be aware. And you might remember that also that you're going to see that acrodynia in Kawasaki disease. You might also have abdominal pain, but the primary organs, again, are going to be the kidney and the brain. So who gets mercury poisoning? Well, some common sources of mercury include certain kinds of fish, so big fish like shark and swordfish, tilefish, and king mackerel. Now, because of the potential harm to the fetus, so you can get some fetal brain damage, the FDA has recommended specifically that pregnant women avoid eating those type of fish, which we mentioned. So the big fish, shark, swordfish, king mackerel, tilefish. So on your test, they might give you a scenario where a patient likes eating exotic fish or returns from a recent fishing trip. They caught some swordfish, and then soon after that, maybe they develop enlarged kidneys, maybe they get some neurological symptoms. So I want you to be able to make that connection to mercury poisoning if you see that. Mercury is also found in old thermometers and in batteries, but the organic mercury that's inside living organisms that you would ingest is much more likely to get integrated into your body and be problematic than the inorganic mercury, which would be the mercury that you might play with if you broke a thermometer up. All right, guys, that brings us to the end of the lecture part. It's time for that end of session quiz. Let's go through those answers together. First question here, what has been the most common clinical scenario of excess iron ingestion in the last 3 decades? Well, this is going to be an infant or someone young, a toddler, maybe consuming the iron-fortified vitamins, especially prenatal vitamins. Next, what are some of the clinical effects of zinc deficiency? Well, there's a ton of things. Things you probably need to remember: delayed wound healing, a decreased immune response. You can have a rash around the eyes, mouth, nose, and nares. That's called that acrodermatitis enteropathica. Zinc deficiency can also cause anorexia, diarrhea, can cause growth retardation in children, depressed mental functioning, impaired night vision, and then zinc deficiency can also cause infertility as well. Next, what are some of the signs of hypocalcemia? So remember, you're going to get that tetany, neuromuscular irritability, where you have a lot of involuntary contractions of the muscles, a lot of spasticity. You can get that Vostick sign where you tap the patient's cheek and you get that facial muscle spasm. And then remember, Trousseau sign is where you tighten a blood pressure cuff in the patient's arm, and after about a couple of minutes, that arm goes into that carpal spasm. You definitely need to know both of those. Then the last question here: what organs are primarily affected by an excess of mercury? Remember, that's going to be the kidneys and the brain. All right, that's going to be it for the quiz. Now we have one rapid fire fact. If you see bluish colored lines on the gingiva, you should probably think about lead poisoning. So these are called Burton's lines. All right, guys, that's going to bring us to the end of Biochem 16. I hope you learned something. I'll see you next time.
Let's go over the warm-up questions. First one says, which cytokine inhibits T helper 2 cells? That's interferon gamma. Next, what dermatologic finding is associated with Bacillus anthracis infection? You can see a black S scar with necrosis surrounded by an edematous ring like you see here. This is caused by lethal factor and edema factor. The last question, what are the common causes of ARDS? Well, ARDS can be caused by shock, infection, toxic gas inhalation, acute pancreatitis, aspiration, heroin overdose, and high concentrations of oxygen for extended periods of time. Now let's get started with the lecture.
425. Oh, hello there. Welcome to our Step 1 video on fat-soluble vitamins and antioxidants. I'm afraid you've caught me doing my pre-lecture workout. I never miss it. For some reason, other lecturers don't commit themselves to 425 curls before each lecture. Where is their commitment? Hello, how can anyone expect to provide high-yield material without ripped pythons? That's just common sense. Ooh, let's talk about vitamins. So vitamins as a whole, pretty decently high yield for step 1. Probably even a 5-star topic. Don't get to see those all the time. So expect to have 2, maybe even 3 questions on vitamins. So first, what are the fat-soluble vitamins? That's what we're going to concentrate on today. Remember ADEK, A, D, E, and K. And also, now that we're thinking about fat-soluble vitamins, it's very important to keep in the back of our minds that any fat malabsorption disease, things like cystic fibrosis or pancreatic insufficiency, can potentially result in deficiencies of these types of vitamins. Also, think of diseases or pathology that might affect the ileum, and that's where a lot of the absorption of these vitamins occur as well. So let's start with vitamin D. Vitamin D is a complex but pretty important vitamin in the body. And though we refer to it as a vitamin, its structure is actually quite similar to steroids, things like testosterone, cholesterol, even cortisol. So first, we humans get our vitamin D by ingesting it in the diet. But also, we can actually synthesize certain forms of vitamin D in the skin when the skin is exposed to sunlight. Now, vitamin D is also manipulated into several different forms, especially in the kidney and the liver. So just saying vitamin D doesn't tell us very much. You need to know the different forms and names of vitamin D. So let's go through those. So first we have ergocalciferol, or D2. This form of vitamin D is mainly just ingested from plants. And then we have the cholecalciferol, the D3. You hear about this one a lot. This is added to things like fortified milk. It's also what is synthesized in our sun-exposed skin. Now let's move on to here. In the liver, vitamin D3 and D2 are converted by the enzyme 25-hydroxylase to something called 25-hydroxyvitamin D, sometimes also referred to as 25-OH vitamin D, or its other name is calcidiol. So lots of names for the same thing. Now this is what is measured most often when we're actually checking a patient's vitamin D status. You're going to get that 25-OH vitamin D. Now then the kidney, another player here, that 25-OH vitamin D is further hydroxylated to 1,25-dihydroxyvitamin D, or calcitriol, and that's going to be by several enzymes, but the most important one being 1-alpha hydroxylase. Now this 1,25-dihydroxyvitamin D, or calcitriol, is the physiologically active form of vitamin D. So now that we know the different names of vitamin D, what does it all do? Well, vitamin D increases calcium uptake in the intestine by increasing the expression of calcium-binding protein. Now, it also works in the intestine to increase the absorption of things like phosphate and magnesium. It's also going to stimulate the parathyroid hormone-dependent reabsorption of calcium in the distal tubules of the kidney as well. And it stimulates the bone reabsorption when necessary along with PTH. So all three of these are going to increase serum calcium. You're getting more calcium from the intestines, more calcium from the kidney, and then also you're getting calcium from the bones themselves. So now let's cover some vitamin D deficiency and some resistance. So this occurs by one of four mechanisms. So first, you can have inadequate dietary vitamin D. You're just not taking it in. You can also have impaired hydroxylation by the liver to make that 25-hydroxy vitamin D. You could also have impaired hydroxylation by the kidneys to make that more active one, that 125-dihydroxy vitamin D. Or you can have, finally, in-organ insensitivity to vitamin D itself. So what are the end results of a vitamin D deficiency or resistance? Well, you get rickets in children and osteomalacia in adults. Now, these are both demineralization problems of the bone. Now, why does this happen? Well, let's think about this. Let's say you don't eat any vitamin D, you're low in vitamin D. Without vitamin D, you can't absorb calcium and phosphate very well from the intestine. We described that before. As the serum calcium level drops, the parathyroid glands start to notice this, and they start pumping out parathyroid hormone. Now, that PTH is going to increase the serum calcium by a little bit by breaking down bone. PTH also works to decrease serum phosphate. It's phosphate trashing hormone is one way to remember that. So not only are you breaking down bone, but you now don't have adequate amounts of calcium and phosphorus or phosphate to produce good, strong bones in the future. So this results in bone demineralization or softening of the bones. Patients will also have bone tenderness, they're going to have muscle weakness, skeletal deformity, so they might get bowing of the legs. You might get growth problems, pathologic fractures, and dental problems as well. Now, one important note is that breast milk does not generally have adequate amounts of vitamin D. So most infants are going to get enough sunlight to counteract this, but it is generally recommended that vitamin D supplementation be given to all infants who are exclusively breastfed. Now, what if you take in too much vitamin D? Now, this doesn't occur by getting too much sunlight. It occurs when patients are taking in too much vitamin D supplementation. Patients may become hypercalcemic, which is the big problem, which may lead to those hypercalcemic problems. Remember the mnemonic stones, bones, groans, psychiatric overtones. Now, one other rare instance of vitamin D toxicity can arise in sarcoidosis. Here we see an increase in the conversion of that 25-hydroxy vitamin D to the 125-dihydroxy vitamin D, that more active form. The macrophages present in granulomas can actually increase this conversion. So patients with sarcoidosis may have hypercalcemia because of this. Okay, since vitamin D is a bit more complicated than the average vitamin, let's hit this again in a Lewis Notes. Hello everyone, welcome to another Lewis Notes. Today we're doing vitamin D, and vitamin D is a little bit tricky, so we're going to go through this real quick as a review. So first we get vitamin D up here at the top. You'll notice that my little pink guy who has a stomach and an arm here, and you're producing, remember, D2 and D3 in the diet, just D3 in the skin if it's being exposed to sunlight. So remember D2, D3, and then those are going to go to the liver. And what enzyme are we using over here in the liver in order to make some changes here? Remember, this is our 25-hydroxylase. So 25-hydroxylase is going to be what's in the liver. And what is that going to produce? That's going to produce your first change. So that's going to be the 25-OH vitamin D, also known as 25-hydroxy vitamin D, also known as calcidiol. So then that calcidiol can then go to the kidneys, and we got this nice purple kidney looking thing over here. And it has several enzymes. Probably the one you got to remember is the 1-alpha hydroxylase. There's also a 24-alpha hydroxylase, but I would probably just remember the 1-alpha hydroxylase is the most important thing here. And that leaves us with that double hydroxylated, that 125-dihydroxy vitamin D, also referred to as calcitriol. So those are going to be the big changes here. So now let's do a quick little thing on vitamin D deficiency. So you might want to use like a red pen or something to kind of show what's going on. So let's say if you have a decreased vitamin D, say you're not getting it up here, down here we're kind of knocking out our vitamin Ds or something, and maybe you never go outside, or you're just not able to kind of help produce as much vitamin D as you want to. So what happens? Well, remember that vitamin D was going to help you in the stomach and in the intestines and all this to produce an increase in calcium. So you're going to get a decrease in calcium from the gut right here. You're also going to get that decrease in phosphate as well. So decreased phosphate, decreased calcium as well when you're knocking out your vitamin D here. So what does that happen? So well, you see that decrease in serum calcium, and then you look over here at your parathyroid. So what is your parathyroid going to do? Well, your parathyroids, when they see that decrease in calcium, they're going to increase PTH. So an increase in PTH is what happens here. And that PTH is going to go where? To the bone. So we go to the bone here, and what is that going to do? Well, that's going to help increase the calcium a little bit. It's going to break it down, though. And remember that PTH can also stand for phosphate trashing hormone. So you're going to decrease phosphate again as a result of that decrease in the vitamin D there. Now, also the other thing in the kidney over here, remember the kidney also responded to vitamin D. So if you have a vitamin D deficiency, you're also going to have a decrease in calcium again in that situation. So vitamin D really important in calcium, very, very important. That's why we always give vitamin D when we're trying to give calcium to people as well to help prevent things like osteoporosis. But if you do end up with a vitamin D deficiency, remember you are going to get things like bone tenderness. You're going to get things like skeletal deformities. You might get bowing of the legs. You might get tooth problems. You might get a lot of different things, growth problems, pathologic fractures, all that type of stuff. So vitamin D, very, very important. A little complicated because it gets switched around a couple different times. But overall, if you can just follow this, I think you'll be okay. All right, so let's move on to vitamin K. So what is the principal role of vitamin K? Well, it's involved in the post-translational modification of various clotting factors. And it's going to serve as a coenzyme in the carboxylation of certain glutamic acid residues present in these proteins. So you have to have vitamin K in order to have normal clotting systems. And it's also interesting that vitamin K is actually synthesized by the intestinal flora. So you have to have a normal intestinal flora to produce adequate amounts of vitamin K. So which clotting factors are involved? Well, they include protein C and S, and then the clotting factors 2, 7, 9, and 10. Now, one mnemonic that we've used for a while is DISCO started in 1972. Not really accurate, but hey, we got to remember this somehow. So 10, 9, 7, 2 for the 1972, and then the S and the C in DISCO. So these are the vitamin K dependent clotting factors, which are also the clotting factors that would be decreased if you were to take something that would inhibit that, like warfarin. Now, another high-yield topic is the relationship between vitamin K and newborns. So newborns can have problems with hemorrhage. And the reason is that newborns have a sterile gut. Therefore, they don't have the appropriate intestinal flora to produce vitamin K. Now, breast milk only provides about 1/5 of the needed vitamin K. Therefore, it's recommended that newborns receive a single IM dose of vitamin K at birth. And that should really carry them over to when they can actually get appropriate bacteria in their gut and make their own. Now, what's going to cause a vitamin K deficiency? Well, we don't have to eat a lot of vitamin K because our gut flora is already producing it. So usually it's a medication problem that's going to lead to a vitamin K problem. So warfarin is a vitamin K antagonist, and we're using this often as an anticoagulant. Other medications, not quite as potent, things like anticonvulsants, phenytoin can cause some of this. And then antibiotics, which would actually wipe out your gut bacteria. Then you would potentially or theoretically not have that endogenous vitamin K production anymore. Now let's move on to vitamin A. So vitamin A is another fat-soluble vitamin that includes several compounds. Things that are included in this: retinol and retinal. So retinol is especially useful to the eye and the immune system, maintenance of the epithelial cells and mucus-secreting cells as well. You can also have something called beta-carotene, which is cleaved in the intestine to yield 2 molecules of retinol. And then we have something called retinoic acid. So retinoic acid actually can't be reduced, so it's unusable by the body, but it's useful for the treatment of certain diseases, and we'll go over that in just a little bit. So all these different types of vitamin A. So dietary vitamin A is important for some normal functions of the body, but when do we use it clinically? Well, most of the time we use vitamin A to treat acne. So tretinoin is a very commonly used topical agent that is used for mild to moderate acne. The oral form of vitamin A that's used for more severe acne is called isotretinoin. Now, these vitamin A drugs act to decrease the size and secretion of sebaceous glands. Now, another treatment worth mentioning is that vitamin A can be useful in the treatment of measles, particularly in third world countries, and can also be useful potentially in the treatment of AML, acute myelogenous leukemia, especially the M3 subtype. Now, what are some of the signs of deficiency of vitamin A? Well, you can have night blindness, so think about a lot of eye issues. Xerophthalmia, which is a pathologic dryness of the conjunctiva, and cornea resulting in corneal ulceration and blindness. So if you see corneal ulceration, you need to at least consider a vitamin A deficiency. You can have chromatomalacia, which is wrinkling and clouding of the cornea. And then you can see something called the Biddix spots, which are dry, kind of silver gray plaques on the bulbar conjunctiva. Now, what are some of the signs of vitamin A toxicity? Well, you can have things like headache, you can have nausea, vomiting, you can have stupor, you can have an increase in intracranial pressure, and we call this pseudotumor cerebri. Now, the skin is going to also potentially get very dry and pruritic. Too much beta carotene can actually cause a kind of benign orange coloration of the skin. So too much beta carotene will not lead to a vitamin A toxicity, but you'll look orange. The liver can become enlarged and possibly cirrhotic with too much vitamin A, and then you can get bone and joint pain. So anyone that's on something like an oral vitamin A like isotretinoin, you actually have to be real careful in monitoring a lot of these symptoms. And alopecia is also one last thing that you need to look for. Okay, sir, in which patient populations is vitamin A supplementation a particularly bad idea? Well, if you're pregnant because of the risk of teratogenesis, you really want to avoid vitamin A. And this is why you always want to ask a woman of childbearing age if she's pregnant or plans on becoming pregnant before you prescribe them any of the forms of vitamin A, especially the oral form. We always get a negative pregnancy test before thinking about starting isotretinoin. There's also other rules in place on who can actually prescribe that medication because teratogenicity is a huge problem with isotretinoin. Let's move on to vitamin E, also known as alpha tocopherol. Now, its primary function is as an antioxidant. It prevents a non-enzymatic oxidation of cell components by oxygen-free radicals, especially on red blood cells. So it's going to actually help prevent free radicals from damaging our red blood cells and then also other cells as well. Now, what is associated with a vitamin E deficiency? So if we're trying to protect ourselves, think of the opposite of that. We might see hemolytic anemia because there's less protection of those RBCs. You might see spinocerebellar degeneration resulting in ataxia. Those cells are being attacked. And then maybe some peripheral neuropathy and proximal muscle weakness as well. All right, so those are going to be our fat-soluble vitamins. Remember D, K, A, E, or EDEC, or ADEC, however way you want to remember it. Now, we said vitamin E is an antioxidant. What other substances in the body are antioxidants? Well, vitamin C is also an antioxidant. So we're actually going to go ahead and talk about vitamin C here, even though it is a water-soluble vitamin rather than a fat-soluble vitamin. But since it's an antioxidant, we're going to cover it right here. So vitamin C is also known as ascorbic acid. Now, what is the main metabolic reaction that vitamin C is involved in? Well, it's the hydroxylation of prolyl and lysyl residues of collagen. So this is very important in the hydroxylation of proline residues and for lysine residues, and that's in order to form a more appropriate collagen production. So if you're deficient in vitamin C, you're not going to be able to make collagen very well, and we'll see in a second what that results in things like scurvy. But there are a few other things that vitamin C does. It's required in the conversion of dopamine to norepinephrine. So dopamine beta hydroxylase actually needs vitamin C in order to make norepinephrine. Additionally, vitamin C circulates in the blood and serves as an antioxidant which helps protect red blood cells. Also, vitamin C facilitates iron absorption in the gut. So vitamin C will help keep iron in its reduced state, which makes it easier for the gut to absorb. And that's why we tell people to take their iron with orange juice. Now, what are the major signs of vitamin C deficiency or scurvy, which is just great because it makes you think of pirates? So you can have sore, spongy gums, you can have loose teeth, you can have fragile blood vessels causing hemorrhages, swollen joints, bleeding into joint spaces, so you get hemarthrosis. You can get impaired wound healing as well, and anemia. All right, guys, that's going to be it for our video on the fat-soluble vitamins plus vitamin C. So let's go ahead and go through the end-of-session quiz together. First question, what vitamin in excess can cause hypercalcemia? So remember, if you're taking way too much vitamin D, that might actually result in a hypercalcemic situation. Next question, what are the symptoms of vitamin A toxicity? Well, chronic toxicity can cause visual impairment, so think of the eyes. But you can also get nonspecific stuff. Fatigue, you might see some ataxia, some headache. You might actually even see increased intracranial pressure. It can also cause dry skin, alopecia, hyperlipidemia, hepatotoxicity, arthralgias. And remember that retinoic acid, highly teratogenic. So you don't want to give something like isotretinoin to women who might become pregnant. Next question. What clinical features would lead you to suspect that a patient has scurvy other than them having a parrot on their shoulder? So if they have sore, spongy gums, if they have loose teeth, if they have fragile blood vessels that are causing a lot of hemorrhages, think of those swollen joints, that hemarthrosis that you might see, impaired wound healing, and anemia. Next, vitamin C is necessary for the hydroxylation of which amino acids in collagen synthesis? So think of proline, think of lysine. Those are the two big ones that they're going to affect. Next, which enzymes are used to convert cholecalciferol to calcitriol? So this is kind of a multi-part section here. So cholecalciferol, that D3, taken in by a diet, also produced by the skin when directly in sunlight. It is converted to the calcidiol by 25-hydroxylase, and that's in the liver. And then that calcidiol is then converted to calcitriol by 1-alpha-hydroxylase in the kidney. All right, guys, that brings us to the end of the end of session quiz. We do have a couple rapid fire facts. Let's go through those. The treatment of choice for rickets or osteomalacia, well, those are just vitamin D deficiency, so you just get them vitamin D. Patient with swollen gums, poor wound healing, bleeding mucous membrane, spots on the skin would make you think about scurvy. And then remember, that's that vitamin C deficiency. All right, guys, that brings us to the end of Biochem 17. I hope you learned something. I'll see you next time.
Let's go over the warm-up review. First question. Describe which light reflexes would be seen in both eyes if the right ocular motor nerve were damaged. So this is known as an efferent defect. The right eye will not respond to light shown in either eye, but the left eye will constrict when a light is shown in either eye. Next, what's the differential diagnosis for eosinophilia? Well, our mnemonic is Canada P, which stands for collagen vascular disease, atopic diseases, neoplasms, Addison disease, drugs, acute interstitial nephritis, and parasites, especially those helminth infections. The last one. What substances act on smooth muscle myosin light chain kinase, and how do these affect blood pressure? So the three substances we discussed are dihydropyridine calcium channel blockers, epinephrine, acting at the beta-2 receptors, and prostaglandin E2. And then all these affect blood pressure by lowering blood pressure. Even epinephrine, when it acts at beta-2 receptors, that causes vasodilation. Now we're ready to start the lecture.
Oh, I'm afraid you've caught me at home. Hello everyone and welcome to our Step 1 video on water-soluble vitamins. Please excuse my casual attire. When I get off from a hard day's work at DIT, I just like to come home and relax, you know, like just hang out with my tigers. This one's named McGinnis. He's not very bright. Now, I've just got to say, you've all done an amazing job studying so far. And because you're such an impressive student, DIT is going to send you a tiger.
No, we're not.
Travis, how did you get in my house?
We don't have any tigers.
What do you mean we don't have any tigers?
We just don't.
Well, just go down to the store and get some.
You can't just buy a tiger at the store.
Sure you can. You know, just talk to the guy, ask him if they have any in the back or something. Put out a little effort, Travis. This might be why you don't have any tigers.
Whatever.
Students, don't worry, I'm all over this. And remember, in life, never settle for anything less than tigers. Okay, so in this video we're going to talk about our water-soluble vitamins, which includes basically all of the B vitamins. It also includes vitamin C, but since we've already talked about vitamin C in some detail with the antioxidants, we're really just going to talk about the vitamin Bs today. So let's start off with vitamin B1. So that's our first vitamin, and it is thymine. Now the active form of B1 is actually thymine pyrophosphate, and actually has a ton of important functions. So think about the conversion of pyruvate to acetyl-CoA. That's going to be catalyzed by pyruvate dehydrogenase. Thymine is a very important cofactor there. Alpha-ketoglutarate to succinyl-CoA. So that's going to be catalyzed by alpha-ketoglutarate dehydrogenase. You can use the mnemonic tender loving care for nobody for the required cofactors for both that pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase. Well, the thymine is the tender, the T in that mnemonic there. Then another one, ribose 5-phosphate to glyceraldehyde 3-phosphate. That's going to be catalyzed by transketolase. And that's actually in the pentose phosphate pathway. And that reaction, again, requires thymine. So very important stuff that thymine does. So you do need to know those enzymes. But even more importantly, you need to know what happens with thiamine deficiency. So a thiamine deficiency occurs in the setting of poor nutrition. So think of an alcoholic patient who's not eating very much. Malabsorption is another big one, or increased water loss due to dialysis. Now, as we saw before, thiamine is extremely important in oxidative metabolism. So if you don't have thiamine, then you can't break down glucose and form ATP. So organs like the brain tend to be affected first with a thiamine deficiency. Also, in the absence of thiamine, there tends to be damage to the medial thalamus and the mammillary bodies of the posterior hypothalamus, as well as generalized cerebral atrophy. So patients can get what's called Wernicke-Korsakoff syndrome. This material is actually covered in the video with alcoholism as well, but we'll cover it here too. Now, Wernicke-Korsakoff can be confusing because it's actually two different syndromes, each representing a different stage of the disease. Wernicke encephalopathy is an acute syndrome. Korsakoff syndrome is the kind of chronic neurological condition that usually occurs as a consequence of that initial Wernicke encephalopathy. So first, what is that Wernicke encephalopathy? Well, the classic triad is encephalopathy. That makes it easy. You get some ocular dysfunction. You also get some gait ataxia. But patients can also have things like stupor, coma, hypotension, and even hypothermia. Now, Korsakoff syndrome, as we said before, is that chronic consequence of having a Wernicke encephalopathy, and it has several symptoms as well. First is memory loss, and that includes a retrograde and an anterograde amnesia. So you can't remember things in your past, nor can you create new memories. Confabulation is another big one. So patients just sort of invent memories that he or she is going to believe that are true. It's actually trying to fill in a lot of those memory gaps. You also see some personality changes, some apathy as well. And just to make things a little more confusing, in addition to Wernicke-Korsakoff, patients can also get what's called beriberi. Now, historically, beriberi is seen in areas where people's primary food source is a polished or dehusked rice. So when you remove the husk, you actually remove the thiamine from the rice. Beriberi is described as either wet beriberi or dry beriberi. Dry beriberi symptoms include things like non-specific peripheral neuropathy with a myelin degeneration. You get toe drop, wrist drop, foot drop, so a lot of drops. You get generalized muscle weakness. You get hyporeflexia and even areflexia. Now, what characterizes the wet beriberi? So you have a lot of peripheral vasodilation, and that actually leads to a high output heart failure. So you get a lot of peripheral edema. You can also have cardiomegaly as well. So dry beriberi is involving the nerves. Wet beriberi is involving the heart. What about treatment? Treatment will involve immediate thiamine supplementation. Now, one very important treatment point is that giving glucose to a thiamine deficient patient can actually worsen the Wernicke encephalopathy. So anyone who is at a risk of a thiamine deficiency should always get thiamine before glucose. Let's move on to vitamin B2. B2, also known as riboflavin. So there are two biologically active forms of riboflavin. So the riboflavin can be flavin mononucleotide, so FMN, and you can also have flavin adenine dinucleotide, so FAD. Now, both of these are cofactors for redox reactions. So if the enzyme has the word dehydrogenase in it, then it likely requires B2 as a cofactor. Now, what are the symptoms associated with riboflavin deficiency? Well, patients will have dermatitis, angular stomatitis right in the corners of your mouth there, glossitis, so kind of a smooth, sometimes red, sometimes purple looking tongue. Let's move on to vitamin B3, also known as niacin. So B3 has two active compounds. There's nicotinamide adenine dinucleotide, so NAD, and then you have nicotinamide adenine dinucleotide phosphate, so NADP. Now, these are important again in redox reactions. Now, you might remember from our lecture on amino acids that these are derived from tryptophan. So you can actually make your own niacin if you have enough tryptophan. Now, what disease is caused by niacin deficiency? Well, it's pellagra. So here you have your 3 Ds of vitamin B3 deficiency. So the 3 Ds are dermatitis, diarrhea, and dementia. Now, vitamin B3 deficiency can occur from a dietary deficiency. Also important to remember here that pellagra can be caused by heart-nut disease. You actually get decreased tryptophan absorption in that situation. You can have malignant carcinoid syndrome. In that case, you actually have increased tryptophan metabolism. And then also a drug, INH, which actually inhibits both B6 and B3. Moving on to vitamin B5. So B5 really isn't as high yield as some of the other B vitamins, also known as pantothenate. So it is a compound of coenzyme A, which functions in the transfer of acyl groups. Now, if deficient, then you can see some dermatitis, some enteritis, which is a little different here. Alopecia is a little new, and then adrenal insufficiency. And that's about all I would know about B5. Moving on to vitamin B6, also known as pyridoxine. So the active form is pyridoxal phosphate. So what is the metabolic function of our pyridoxal phosphate? Well, it's a coenzyme for numerous enzymes, including those of amino acid metabolism. So think about transaminations and deaminations. B6 is also very important in converting amino acid precursors into a lot of stuff. Things like into heme, niacin that we just talked about, histamine, GABA, dopamine, norepinephrine, epinephrine, a lot of stuff. Now, what are the clinical findings of B6 deficiency? Well, you'll see many of the same symptoms as riboflavin deficiency, things like that angular stomatitis around the lips, the glossitis of the tongue as well. But one thing that's unique to B6 deficiency is things like convulsions or seizures and hyperirritability and peripheral neuropathy. And those convulsions occur because B6 is actually essential, as we said before, to the creation of GABA. And that's the main inhibitory neurotransmitter of the brain. GABA keeps the brain quiet. Now, as we just saw with vitamin B3, INH or isoniazid can induce a deficiency of B6. So if a patient is receiving treatment for tuberculosis, then we often add on vitamin B6 as another treatment option. Moving on to vitamin B7. B7 is biotin. So what's our metabolic role here? Well, it's an apoenzyme for carboxylation reactions. So if you want to add CO2 to something, then you have to use biotin. Also, if you see an enzyme with the word carboxylase, that's another clue in there. It's going to require your B7 or your biotin as a cofactor. Now, what can cause a deficiency of biotin? Well, there's a glycoprotein called avidin, and that's found in egg whites, and actually prevents absorption of biotin. You actually have to eat quite a few egg whites for that to take place, but that's still pretty interesting. Antibiotic use can also result in a biotin problem as well. Moving on to vitamin B9, folic acid. So folic acid, big one here. Simply put, folic acid is crucial for the synthesis of DNA and the repair of DNA. Also especially important in the synthesis of purines and pyrimidines. Folate is also very important for rapid cell division and growth. Its biologically active form is the tetrahydrofolate, which is a coenzyme for one carbon transfer or methylation reactions. Now, humans are actually unable to synthesize their own folic acid, therefore we have to get our supply through the diet. Now, let's get into the fun stuff. What about folic acid deficiency? How does that occur? Well, most of the time, folic acid deficiencies are due to medications or malnutrition. Drugs like phenytoin, sulfonamides, trimethoprim and methotrexate can all cause a folic acid deficiency. Now, what's nice is that you can actually counteract these effects by just giving supplemental folic acid in most cases. Now, folic acid can also be low in pregnancy because of that rapid growth of the fetus. And the fetus is requiring a lot of that folic acid to rapidly divide its cells. But what are the consequences, again, of that folic acid pregnancy, especially in pregnancy? Well, if the folate deficiency happens in utero, then the baby might develop a neural tube defect. And folate deficiency is the most common cause of neural tube defects, and that's why it's very important to supplement that folate before a woman actually plans to become pregnant. Now, as an adult and also as a child with folate deficiency, you can have a megaloblastic anemia because folate is essential to rapidly dividing cells. Then there are some other non-specific findings, glossitis, similar with our other things, diarrhea, depression, and confusion. I want to take just a very brief moment and talk about megaloblastic anemia here. So this is an anemia, but the RBCs are very large. You have a type of what we call macrocytic anemia, which means large red blood cells. And this anemia is characterized by an elevated number of megaloblasts in the marrow. Now, under the microscope, you might often see hypersegmented neutrophils and then again those large RBCs. Now, including our folate, what's the other vitamin that can lead to a megaloblastic anemia? That's going to be B12, and that's one we're going to hit here in a second. So B12, also known as cobalamin. And B12 is, again, a very important vitamin because B12 is a cofactor with homocysteine methyltransferase to convert homocysteine to methionine. Also in the process, a tetrahydrofolate is produced. Where did we just see that? Remember, that's the active form of folate, so they are related there. Also, the conversion of methylmalonyl-CoA, MMA, to succinyl-CoA requires B12 as well. So what are we going to see with a B12 deficiency? Well, just like with our folate, we just talked about megaloblastic anemia, those hypersegmented neutrophils. Patients can also have neurological symptoms, including paresthesias, ataxia due to loss of vibration and position sense, memory loss, dementia, severe weakness. Now, many of these symptoms seem to be linked to a defect in myelin formation that occurs with that B12 deficiency. Now, this is another actually big differentiating characteristic from folate deficiency. Both B12 and folate, as we talked about before, causes that megaloblastic anemia, but only B12 deficiency will have neurological symptoms. So how does one become deficient in B12? Well, first you need to know that B12 is only found in animal products. So strict vegetarians or vegans can actually have a problem with low B12 if they're not supplementing. Next, you need to know how B12 is absorbed, because the most common cause of B12 deficiency is improper absorption. Now, B12 is found to intrinsic factor in the duodenum, and then that complex together is then absorbed in the terminal ileum. Now, the intrinsic factor is produced by parietal cells in the stomach. So an important cause of B12 deficiency is what's called pernicious anemia. So this is an autoimmune destruction of gastric parietal cells and antibodies against intrinsic factor itself. Now, without those parietal cells, the body can't produce intrinsic factor, and without intrinsic factor, B12 cannot be absorbed in the ileum. Now, a similar scenario can occur with gastric bypass surgery. If you remove enough of the stomach, then you decrease the amount of intrinsic factor as well. Now, other causes of B12 deficiency include malabsorption problems like Crohn's disease, celiac sprue, and enteritis. Anything that can cause interference in the terminal ileum can impede the absorption of that B12 intrinsic factor complex. So how do you treat a B12 deficiency? Well, if you're able to absorb B12, then you can just give oral supplements. But we'll probably at least start with an IM dose of B12 to get people started. Now, how would you know if this B12 deficiency was from pernicious anemia? Well, you can check for antibodies to that intrinsic factor. That was one of the pathological problems there. Also, you can perform what's called the Schilling test. So this is where you actually give a patient radio-labeled B12, and then you test to see how much shows up in the urine. If the urine B12 is lower than you should expect, then you might have an absorption problem because it's not going through your body. All right, so before we get to that end of session quiz, let's do another Lewis Notes because, man, that is a lot of material. Hello everyone, welcome to another Lewis Notes. We're going to cover these vitamins again. We're just looking at some of the high points again, not every detail, but enough to remember some of the high yield stuff. So let's go through these. First, over here on the right, we're going to be taking these and matching them again. So first one, we have cofactor with homocysteine methyltransferase. So this is a hard one to remember, but this is actually vitamin B12. So B12 is going to go over here. Remember with B12, you're going to get that megaloblastic anemia, that peripheral neuropathy. Next, we have NAD, NADP on redox reactions. So that's going to be your B3. That's going to go right there. Component of coenzyme A. So that's going to be vitamin B5. That one's hard for me to remember. B5 is not as commonly thought of. And if you get a deficiency, you're going to get some dermatitis, some enteritis, some alopecia, maybe even some adrenal insufficiency. Next, we have good old-fashioned Wernicke encephalopathy. That one's relatively easy. That's going to be your thiamine deficiency, your B1. Think of Korsakoff syndrome as well with that one. Next, we have production of heme, niacin, and GABA. You could also put in there histamine and dopamine and even norepinephrine and epinephrine. That's going to be vitamin B6. Remember that you're going to get a deficiency of that potentially with tuberculosis treatment. So if you're using INH, you also need to use B6 because they can lose that B6 in that medicine right there. Next, we have FMN, FAD, redox reactions again. So redox reactions, when we say that, we mean dehydrogenase is a good example of that. So that's going to be your B2. And then with B2, you might get a dermatitis, angular stomatitis, some glossitis as well. Another hard one for me to remember is carboxylation reactions. That one's just a memorization thing here. That's going to be B7. Nothing else too interesting with that one. And then finally, we have synthesis of purines and pyrimidines. That's going to be your B9. That one's also important with DNA repair. And you might see this with deficiency with drugs, things like phenytoin, sulfonamides, trimethoprim, methotrexate as well. So that's folic acid. So in pregnancy, you're going to see neural tube defects. You're also going to see some megaloblastic anemia as well. All right, guys, those are some of the high points for our B vitamins. I'll see you next time. All right, it's time for another end of session quiz. Try to answer those questions on your own and then we'll go through them together. First question, a patient presents with convulsions and irritability. What vitamin deficiency is causing these symptoms? So the thing you want to think about is that convulsions, think about vitamin B6 because that's the one that produces the GABA. Next, what type of anemia can be caused by folate or B12 deficiency? So that's that megaloblastic anemia. Next, where is B12 absorbed into the circulation? So you got to think about the terminal ileum. Remember, that's that complex of B12 and intrinsic factor. Next, which vitamin deficiency matches each of the following descriptions? So first we have peripheral neuropathy and glossitis. Think of B12, also think about B6. Neural tube defects, that's going to be your folic acid or folate. Dermatitis, diarrhea, dementia, those are the big 3 Ds. Remember, that's B3 or niacin deficiency, which is also referred to as pellagra. Next, we have megaloblastic anemia. We've touched on this many times. Remember, it's going to either be folate or B12 deficiency. Pernicious anemia, remember, that's just specific for B12. And then last question here, which vitamin matches the following statement? So first we have used in oxidation reduction reactions. That's going to be vitamin B2 riboflavin. And remember, that's necessary to make that FAD and that FMN. And then also think about vitamin B3, niacin, which is used to make that NAD or NADP. Next, we have used in carboxylation reactions. So this is Biochem. Now, we didn't talk about this other one. Actually, vitamin K is actually a cofactor for gamma carboxylation of glutamate. But think about biotin as the answer to that question primarily. Next, requires intrinsic factor for absorption. That's B12. Used by pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase. That's going to be primarily B1 thymine. Next, we have deficiency can be caused by isoniazid use, so primarily B6, but don't forget about B3 niacin as well. Next, we have cobalt is found within this vitamin. So we actually didn't talk about this one in the lecture, but it's vitamin B12. B12 is also called cobalamin, so that association shouldn't be too difficult. Next, we have critical for DNA synthesis. I want you to think about folate and B12. All right, that's it for the end of session quiz. We do have a couple rapid fire facts. First we have here is most common vitamin deficiency in the U.S. That's going to be folate deficiency. If you see hypersegmented neutrophils, that's a feature of megaloblastic anemia from either B12 or folate deficiency. And then we have dilated cardiomyopathy, edema, and polyneuropathy. That's going to be wet beriberi, which is due to thymine or B1 deficiency. All right, that brings us to the end of the video. I hope you learned something. I'll see you next time.